नमस्कार, योग पर हमारे नए कार्यक्रम में आपका बहुत बहुत स्वागत है। अपने पहले एपिसोड में हम आपको योग की उत्पत्ति के बारे में बताएंगे।
हम सब ने योग के बारे में तो जरूर सुना होगा, लेकिन क्या आप जानते हैं कि योग हमारी रोजमर्रा की जिंदगी का हर भाग बांधता है? आप पूछेंगे कैसे? बहुत आसान है। योग का अर्थ होता है जोड़ना।
योग युग धातु से लिया गया है, जिसका मतलब होता है टू यूनाइट, यानी जोड़ना। और योग का मतलब है जोड़, समायोजन, हार्मनी, कम्युनियन ऑफ आत्मा एंड परमात्मा।
भारत में बहुतों का मानना है कि 4 वेदों और 6 दर्शन शास्त्रों में हमारी जिंदगी के हर पहलू से जुड़ी दुनिया भर की महत्वपूर्ण ज्ञानवर्धक बातें मौजूद हैं। इन 6 दर्शन शास्त्रों में से आज दुनिया भर में योग का स्थान एक प्रमुख व्यावहारिक ज्ञान का है।
ये हमारा जो पूरा भारतीय वाङ्मय है, इसमें हम 6 दर्शनों की चर्चा करते हैं और हमारे 6 दर्शन है पूरे विश्व के लिए, ये मानवता के लिए है। और पहला दर्शन है हमारा पूर्व मीमांसा है, उसके बाद है उत्तर मीमांसा, उसके बाद आता है न्याय। न्याय दर्शन के प्रवर्तक है गौतम मुनि। न्याय के बाद है वैशेषिक, इसके प्रवर्तक ऋषि है कणाद। और आगे सांख्य दर्शन है। सांख्य महर्षि कपिल का दर्शन है। और छठा दर्शन योग दर्शन है, जो महर्षि पतंजलि का है। और ये जो 6 दर्शन है, ये संपूर्ण भारतीय मनीषा के प्रतीक है।
इसके संस्थापकों ने इसे इस प्रकार परिभाषित किया है कि ये शब्दों, विचारों और कर्मों को इस तरह अनुभव करता है जिसके परिणाम स्वरूप हमें स्वस्थ, अत्यंत आनंदमयी और शांत जीवन की प्राप्ति होती है। हमारे वेदों जैसे महाभारत के मुताबिक आत्मा को इंसान की योनि में आने से पहले 84 लाख बार जन्म लेना होता है और इस तरीके को दरकिनार करने का एकमात्र रास्ता निर्वाण की प्राप्ति है। इसकी प्राप्ति के लिए मनुष्य के रूप में जन्म बहुत अच्छा अवसर है। इसे सुगम बनाने के लिए आश्रमों का निर्माण किया गया, जो है ब्रह्मचर्य, गृहस्थ आश्रम, वानप्रस्थ और संन्यास। ये चारों आश्रम उन लोगों के लिए उन्नति का मार्ग सिद्ध हुए जिन्होंने निर्वाण को प्राप्त किया। आमतौर पर यह माना गया कि जिन्होंने सच्ची धारणा से इन आश्रमों का पालन किया, ज्यादातर वो ही निर्वाण को प्राप्त कर सके। अगर हम इस तथ्य के स्वरूप को देखें तो असल में योग जीवन का ऐसा रूप था जिसको अपनाने की सलाह लोगों को दी जाती थी।
आम लोग ही नहीं बल्कि बड़े बड़े वैज्ञानिक और डॉक्टर भी इसके प्रशंसक हैं। इसका कारण क्या है? इसका मुख्य कारण यही लगता है कि आपकी शारीरिक व्याधियां हों अथवा मन की शांति प्राप्त करने की बात हो, अथवा अंदर की जो खोखलापन है, हमारा भौतिकवाद का खोखलापन है, अशांति है, असंतोष है, क्षोभ है, उससे पार जाने की बात हो, आध्यात्म के रास्ते को चलने की बात हो, योग आपका जीवन के हर पहलू में बड़ा ही साथ निभाता है।
विचार करने योग्य बात यह है कि योगा रात भर में उत्पन्न नहीं हुआ। ये सदियों से चला आ रहा है और कई ऋषियों ने अलग अलग तरीकों से योग का अभ्यास किया और प्राचीन समय से ही योग पर बहुत सी किताबें लिखी जा रही हैं।
इसके अनेक रास्ते योगियों ने बतलाए हैं, जैसे राजयोग, भक्ति योग, ज्ञान योग, लय योग, कुंडलिनी योग, अनेक अनेक रास्ते हैं, हठ योग। लेकिन जब तक इंसान का शरीर ठीक नहीं रहता है, तब तक कोई भी ये जो रास्ते बताए हैं योगियों ने, कोई भी रास्ते के ऊपर चलना इतना आसान नहीं रहता।
जैसा कि बहुत से लोग मानते हैं, योग सिर्फ आसन नहीं है। इसमें से महर्षि पतंजलि का अष्टांग योग ज्यादा प्रचलित है। लेकिन अष्टांग योग क्या है? अष्टांग योग में ऋषि पतंजलि ने योग के 8 अंगों का विवरण किया है, इसीलिए इसे अष्टांग कहा गया है। यह 8 अंग मिलकर व्यवहारिक विधियां होती हैं जो योगी को आसन, ध्यान और साधना के साथ साथ अपनानी होती है। ये है यम या सिद्धांत, नियम या व्यक्तिगत अनुशासन, आसन या योग मुद्राएं, प्राणायाम ये है श्वास की योग क्रियाएं, प्रत्याहार इंद्रियों का संहार, धारणा या एकाग्रता, ध्यान या चिंतन और आखिर में समाधि, मुक्ति और मोक्ष की प्राप्ति। योग के 8 अंगों को आगे और विभाजित किया गया है उन नियमों या विधियों को सम्मिलित करने के लिए जिन्हें योगी को अवश्य अपनाना चाहिए। इनमें से बहुत सी व्यवहारिक विधियां शायद आज के समय में ज्यादा उपयोगी न हो। जैसे अगर हम यम के सिद्धांतों पर नजर डालें जो एक व्यक्ति को समाज में रहते हुए अपनानी चाहिए, तो इसमें आते हैं अहिंसा या हिंसा न करना, सत्य या सच बोलना, अस्तेय या चोरी न करना, ब्रह्मचर्य, आत्मनियंत्रण यानी इंद्रियों का दमन और अपरिग्रह, जितनी जरूरत हो उतना ही लेना। जहाँ तक इन सभी सिद्धांतों को अपनाने की बात है, तो आज की तारीख में तो ये असंभव ही है, क्योंकि ऐसा व्यक्ति मिलना तो मुश्किल है जिसने कभी झूठ ना बोला हो, ब्रह्मचर्य का पालन करता हो और सिर्फ उतना सामान इकट्ठा करे जितना उसे जरूरत हो। व्यक्तिगत अनुशासन भी इससे अछूता नहीं है और इसमें इन सबको सम्मिलित किया गया है। शौच यानी शुद्धि, संतोष यानी तृप्ति, तप यानी सहनशीलता, स्वाध्याय यानी स्वयं का अध्ययन और ईश्वर परिधान यानी समर्पण। इन सभी व्यावहारिक विधियों के पीछे कारण था कि मनुष्य सिद्धांतवादी और सहज जीवन का व्यापन करे, चाहे वो समाज में रहे या जंगल में। उनका ये भी विश्वास था कि मस्तिष्क की शुद्धि से अंत यानी संन्यास का मार्ग सुनिश्चित होता है और इसीलिए ये निर्वाण की प्राप्ति का पहला कदम भी माना जाता है। आज के युग में हालांकि एक विवाहित स्त्री या पुरुष जो योग का अनुसरण करना चाहता है, उसके लिए असंभव होगा कि वह ब्रह्मचर्य का पालन करे या कामकाजी व्यक्ति किसी दूसरी स्त्री या पुरुष से बात न करे, लेकिन इस सब सिद्धांत के पीछे की धारणा को अवश्य अपनाया जा सकता है। इससे फिर से मस्तिष्क और कर्म की शुद्धि पर जोर दिया गया है। एक बार इसकी प्राप्ति हो जाए तो दूसरे स्तर के योग का अभ्यास उतना ही आसान हो जाता है। यम और नियम के बाद आते हैं आसन, जो मूलतः योग आकृतियां होती हैं जो शरीर और मस्तिष्क को आराम और संतुलन देने में सहायक होती हैं।
महर्षि पतंजलि के अनुसार आसन की परिभाषा ऐसे है, स्थिर सुखम आसन। जो व्यक्ति स्थिर होके एक विशिष्ट पोस्चर में एक आसन में लंबे समय तक बैठ सके बिना किसी कष्ट के, उसको आसन कहते हैं।
योग अभ्यास करते समय व्यक्ति को थकान नहीं होनी चाहिए। आसन के साथ कभी कभी लोग प्राणायाम का भी अभ्यास करते हैं, परंतु प्राणायाम क्या है? प्राणायाम श्वास ग्रहण करने और उसे छोड़ने की क्रियाएं हैं। योग के इस अंग में हम श्वास बढ़ाने और नियंत्रित करने का अभ्यास करते हैं। धीरे धीरे जब हम श्वास पर पूरी तरह नियंत्रण कर लेते हैं, जो मूलतः एक अनैच्छिक शारीरिक क्रिया है, तब हम योग के अगले स्तर के लिए तैयार होते हैं, जो ध्यान से शुरू होकर समाधि द्वारा मोक्ष मुक्ति तक पहुंचता है। ऐसा इसीलिए क्योंकि हम उपस्थिति के एक पहलू पर ध्यान देते हैं और अपने दिमाग को दूसरे कोलाहल से मुक्त कर देते हैं। प्राणायाम के बाद योगी अगले योग स्तर पर पहुंचता है, जो है प्रत्याहार यानी इंद्रियों का संहार। इससे मस्तिष्क की ताकत बढ़ने में मदद मिलती है। प्रत्याहार योग का अन्य उच्च स्तर है और इस समय तक योगी जिसने इस कला में महारत हासिल कर ली है, अब लंबे समय तक ध्यान में बैठ सकता है। प्रत्याहार का अभ्यास करते वक्त वह दुनिया से अलग हो जाता है। अगला स्तर होता है धारणा, उसके बाद ध्यान और अनंत समाधि। इन क्रियाओं के बीच में तकनीकी अंतर होता है और निर्भर करता है योगी के ध्यान के स्तर पर। जो योगी धारणा का अभ्यास करता है वो एक वस्तु पर लंबे समय तक ध्यान एकाग्रित करने में निपुण हो जाता है, जबकि ध्यान के समय योगी दिमाग को बाहरी वस्तुओं से अलग करके एक चिन्ह पर चित्त केंद्रित करके उस पर ध्यान करता है। योगी जो समाधि तक पहुंच चुका है, खुशी व परम आनंद की स्थिति में होता है और व्यक्तिगत चेतना को विश्वव्यापी चेतना में सम्मिलित करता है। जीवात्मा और परमात्मा का मिलन, ब्रह्मांड की अनुभूति करना और ईश्वर का प्रत्यक्षीकरण होना ही मनुष्य जीवन की अंतिम उपलब्धि है और यही वो अंत है जिसे पाने के लिए प्राचीन काल से योगी इस कठोर अष्टांग का अभ्यास करते रहे हैं।
मेरे विचार से सभी 8 अंगों का पालन करने का एटलीस्ट प्रयत्न तो करना ही चाहिए। जो मूलभूत इसके सिद्धांत हैं, टू अचीव दैट कम्युनियन ऑफ जीवात्मा विद परमात्मा, वो तो वही रहेंगे। यू कांट चेंज दम। ऋषि हमारे जो महर्षि पतंजलि जी ने जो कहा है वो गलत नहीं कहा, लेकिन आप कहते हैं कि ये संभव नहीं है, कठिन है, लेकिन असंभव मैं नहीं मानता।
इसीलिए जो व्यक्ति इस कला को अपनाने के इच्छुक हैं, वो ऐसा कर सकते हैं योग के किसी एक पहलू से जुड़कर, जैसे आसन या ध्यान, या कोशिश करें कि संपूर्ण जीवन योग के अनुसार ही जिएं। हालांकि आमतौर पर यह माना जाता है कि दिमाग, शरीर और आत्मा के बीच में संतुलन बनाने और एक तनाव मुक्त और रोग मुक्त जीवन जीने के लिए व्यक्ति को चाहिए कि योग अभ्यास उसकी सच्ची धारणा के साथ किया जाए। वैसे तो आज के युग के अनुसार इसे ढाला भी जा सकता है।
तो ये थी हमारी पहली श्रृंखला योग के ऊपर। हम आपसे फिर जल्द मिलेंगे, तब तक के लिए नमस्कार।
Welcome to the session on Organization of Knowledge in Libraries, Library Classification. In this session, we will discuss normative principles, canons for work in the idea plane. Dynamic theory of classification. Theory of library classification developed through the process of formulation and refinement. R.S. Parkey identified two distinct stages in its development through the passage of time. Stage 1 was called by him the descriptive theory because it was the description of the practices followed in library classification. These practices were regarded as the norms for designing classification schemes. It held the sway until early 1950s. The classification schemes designed before 1950s were not based on any theory but merely on either the flair of the designer or his practices in the libraries. The stalwarts who contributed to the descriptive theory through their writings and enunciation of principles were J.D. Brown, E.C. Richardson, E.W. Hume, W.C. Berwick Sayers, Henry Evelyn Bliss, and Dr. S.R. Ranganathan. Some of them also successively used these principles in developing their classification schemes. At the second stage in the development of classification theory, we can see that a sound theory capable of carving out a methodology for the design of classification schemes was originated. This theory was developed by Dr. S.R. Ranganathan between 1948 and 1955. Parkey named the theory in this stage as dynamic theory of library classification because it alone can provide for the degree of detail required by the growing complexity of subject treatments in documents. The dynamic theory was presented for the first time in the second edition of Prolegomena to Library Classification 1957. The third edition of Prolegomena, published in 1967, contained a more advanced version of the theory. Formulation of the descriptive theory to organize knowledge was based merely on the existing practices in classification. As the practices then were independent of any theory, it was the practices that guided the theory. This trend was later reversed. In stage 2, the theory of classification reached a point where it could mold the practice and influence the continued development of the classificatory practices. The dynamic theory was no longer descriptive. It has provided a stable methodology for designing schemes of classification. It helped the designers of library classification schemes to keep pace with the problems of the growing universe of knowledge and make more stable schemes. It also helps the classifiers to find appropriate solution to their problems in classificatory work. Dr. Ranganathan strongly advocated that the subjects should be placed in the schemes of classification in a most helpful sequence. The earlier schemes were based purely on enumeration and hence they failed to observe such a helpful sequence. He therefore rejected the earlier schemes and worked towards developing a theory that could provide a filiatory sequence of classes in a scheme of classification. However, he visualized a perennial problem in this context, and that was the problem of transferring the multidimensional universe of knowledge into a unidimensional or scalar one. He worked out a successful solution to this complicated problem of mapping by means of developing the concept of facet analysis and fundamental categories. The significant characteristics of Dr. Ranganathan's dynamic theory are: 1. it has formulated a set of normative principles to make the work of classification and the designing of classification schemes easier and more systematic. 2. It recognized 3 planes of work in classificatory process. These 3 planes are idea plane, verbal plane and notational plane. Significance of normative principles in classification. The normative principles enunciated by Dr. Ranganathan have greatly contributed to the evolution of the dynamic theory. These principles give a scientific base for library classification. These are useful for both classificationists and classifiers. They serve the following functions in classification. 1. They serve as the basis for the compilation of classification schemes. 2. They help us in the critical study of a classification scheme. 3. They help us in the comparative study of the different classification schemes. 4. They help the classifiers in their day-to-day work, such as construction of a class number for a new subject. Dr. Ranganathan recognized the following 4 levels of normative principles. First one, basic laws. Principles that govern the basic process of thinking. Dr. Ranganathan formulated 6 basic laws, namely law of interpretation, law of impartiality, law of symmetry, law of parsimony, law of local variation and law of osmosis. Second, fundamental laws. Principles that relate to the whole of a discipline. 5 laws of library science formulated by Dr. Ranganathan are considered as the fundamental laws of the discipline library science. These laws are useful in every branch of library science. The 5 laws are: 1. Books are for use. 2. Every reader his or her book. 3. Every book its reader. 4. Save the time of the reader. 5. Library is a growing organism. Third, canons. Principles that relate to main divisions or branches of the discipline, such as classification or cataloging in the case of library science. 4. Principles and postulates. Principles that govern some specific aspect of work within a branch, such as principles for facet sequence and postulates of fundamental categories. The normative principles at the earlier levels are invoked when 2 or more principles at the later level lead to conflicting or equally valid different decisions. 3 planes of work. Dr. Ranganathan also introduced 3 planes of work, namely idea plane, verbal plane and notational plane in library classification. Idea plane deals with concepts viewed by themselves independent of the terms denoting them and the digits or numbers representing them. Verbal plane is the plane of expression of concepts in natural language. It is a plane of words and phrases. Notational plane is a plane of numbers and symbols representing the concepts. For example, a class or subject in the idea plane is denoted in the verbal plane by its name in a particular language and represented by a class number in the notational plane according to a particular classification scheme. Canons of classification. Dr. Ranganathan formulated 43 canons for classification in conformity with his basic laws and laws of library science. These canons are normally used for the design of a scheme of library classification. He categorized them into 3 groups according to 3 planes of work. Thus we have 1. Canons for idea plane 15 numbers. 2. Canons for verbal plane 4 numbers. 3. Canons for notational plane 24 numbers. Canons for idea plane. The work in the idea plane is the most important in the classification. Here the concepts or ideas are arranged in a most helpful sequence on the basis of certain guiding principles. In this context, Dr. Ranganathan identified 5 inherent factors in the arrangement of ideas. They are characteristics, succession of characteristics, array of classes, chain of classes, filiatory sequence. Corresponding to these 5 factors, the 15 canons for idea plane were further grouped into 5. Canons for characteristics 4 numbers, canons for succession of characteristics 3, canons for array 4, canons for chain 2, canons for filiatory sequence 2 numbers. Canons for characteristics. Characteristic is an attribute or quality possessed by an entity. A universe of entities can be classified on the basis of certain attributes possessed by the entities. Only those attributes which help to differentiate an aggregate of entities into 2 or more groups can be called a characteristic. The characteristics selected for division should be easily ascertainable, relevant and permanent. The 4 canons for characteristics deal with the selection of characteristics and the process of division of knowledge. 1. Canon of differentiation. This canon states that a characteristic used as the basis for the classification of a universe should differentiate some of its entities, that is, it should give rise at least to 2 classes or ranked isolates. If we classify the universe of boys, height, weight and intelligence differentiate them into a number of classes. But possession of head cannot differentiate them. Therefore, height, weight or intelligence can be used as characteristics for division of the boys. Possession of head cannot be used for the division. Second, canon of relevance. A characteristic used as the basis for the classification of a universe should be relevant to the purpose of classification. For example, physical strength, height and age are relevant characteristics for grouping the students in class for sports and games. But other characteristics such as intelligence, complexion are not relevant for this purpose. Third, canon of ascertainability. A characteristic used as the basis for the classification of a universe should be definite and ascertainable. For the classification of a given universe, a number of characteristics may be relevant, but some of them may be ascertainable, whereas some others may not be easily ascertainable. We should choose definitely ascertainable characteristics. For example, if the date of death is chosen as a characteristic to divide the scientists in a country, it is not an ascertainable characteristic in the case of living scientists. However, if the date of birth is chosen for the same purpose, then it is ascertainable for all the scientists. 4. Canon of permanence. The characteristic used as the basis for the classification of a universe should continue to be unchanged so long as there is no change in the purpose of classification. The periodicals in libraries can be classified into 2 groups as periodicals published by learned societies, periodicals not published by learned societies. Since the sponsoring of periodicals may undergo changes, the characteristic of sponsorship in publication is not a permanent one. a periodical published by a learned society may be taken up by a commercial agency and vice versa. If we classify the periodicals by their subject, it will be a permanent classification. If we use a characteristic which is not permanent for classifying a particular universe, it is likely that the members of one group may go to another group when there is a change of situation. Canons for succession of characteristics. Characteristics are used in succession one after another to derive the classes and subclasses in the process of classification of knowledge. While doing so, the scheme of classifications should satisfy these canons. First one, canon of concomitance. This canon states that no two characteristics used in succession in a scheme of classification should be concomitant. That is, they should not give rise to the same array of subjects or isolate ideas. Independent characteristics should be applied in succession to give rise to different set of subordinate classes in each stage of division of a universe. For example, age and date of birth are concomitant characteristics because they will not give different subgroups in successive classification of a universe of people. Second, canon of relevant succession. According to this canon, the succession of characteristics in the associated scheme of characteristics should be relevant to the purpose of classification. For example, to classify literature books, Melville Dewey has chosen the sequence of characteristics: language, form and period. Dr. Ranganathan has adopted the sequence language, form, author and work. Both the sequence provide helpful arrangement of books on the shelves. Hence, both are relevant successions for classifying literature books. Third, canon of consistent succession. This canon says that the succession of characteristics should be consistently adhered to so long as there is no change in the purpose of classification. This means that the sequence in the application of characteristics once used should be followed consistently. Otherwise, arrangement of classes will become chaotic. However, if the purpose of classification gets changed, the succession of characteristics may also be changed to suit the new requirements. Suppose a classification scheme follows the sequence of disease and organ, and another scheme follows organ and disease sequence to classify the subject medicine, then it is necessary that these two schemes should follow the particular sequence they have chosen throughout the schedule. Canons for array. Each array of classes in a scheme for classification should satisfy the four canons. First one, canon of exhaustiveness. This canon states that the classes in an array of classes and the ranked isolates in an array of ranked isolates should be totally exhaustive of their respective common immediate universe. When an array of classes are formed from an original universe by applying certain characteristics, every entity in the original universe should find a place in one or the other classes in the array. When a new entity is added to the original universe, it should also be assigned to any of these subordinate classes. Second one, canon of exclusiveness. According to this canon, the classes in an array of classes and the ranked isolates in an array of ranked isolates should be mutually exclusive. This canon implies that the classes forming an array should not overlap each other. An entity should be shared by two or more classes. This canon can be satisfied if all the classes are formed from their immediate universe on the basis of a single characteristic. If two or more characteristics are applied simultaneously to derive a classes in an array, it may lead to cross classification. An entity in the universe may appear in two or more classes in the array. The simultaneous occurrence of an entity in two classes, sharing of two classes by an entity, can be avoided if the characteristics are applied in succession and the classes are kept in different arrays. It is also possible to combine characteristics and enumerate the classes in a single array called coalesced array without violating the canon. A coalesced array is an array formed by putting the various arrays formed at any stage in the progressive classification in succession to a single array, the succession of the arrays being such that the classes fall in the filiatory sequence. This explanation regarding the canon can be made clear by the following illustration. Suppose we group the literature books by two characteristics, by country of origin or by form. We get two arrays: American books, British books, French books, German books by country, poetry books, drama books, fiction books, essays by form. If the two characteristics are applied simultaneously to form an array, a book on American poetry can be placed in two classes: American books or poetry books. This is the violation of canon of exclusiveness. According to the canon, this book should be placed only in one class. Violation of the canon can be avoided if the two characteristics are applied in succession for grouping and the classes are kept in two different arrays as shown on the screen. These two arrays can be combined into coalesced array as literature books, poetry, drama, fiction, essays, American literature, American poetry, American drama, American fiction, American essays, British literature, British poetry, British drama, British fiction, British essays, German literature, German poetry, German drama, German fiction, German essays, French literature, French poetry, French drama, French American fiction, French essays. Third one, canon of helpful sequence. According to this canon, the sequence of the classes in an array of classes and of ranked isolates in an array of ranked isolates should be helpful to the purpose of those for whom it is intended. Dr. Ranganathan says that this canon should be followed in each array and also in any coalesced array in a classification scheme. Helpfulness of a sequence in an array may be determined with the help of few principles such as principle of later in time, principles of later in evolution, etc. Another important aspect here is that helpfulness will vary according to the type of users. What is helpful for one purpose may not be helpful for another purpose. Therefore, a general classification scheme has to be consistent in its sequence. In other words, the scheme should be designed to suit the purpose of majority user group. If a particular library has to meet the requirements of different user groups having different purpose, it has to form separate collections without altering the sequence in the classification scheme. Fourth one, canon of consistent sequence. This canon states that whenever similar classes or ranked isolates occur in different arrays, their sequence should be parallel in such arrays. Wherever insistence on such parallelism does not run counter to other more important requirements. According to this canon, a classification scheme should follow the same sequence in all similar arrays. Adherence to the canon has the following advantages. 1. It reduces the load on memory both for the classifier and the user. 2. It secures economy of time and effort in classification. Consistent sequence in arrays can be achieved in the following ways. First one, by using some principles of helpful sequence. Second, by using one and the same schedule, such as schedule of common isolates, to form similar arrays in different subjects. 3. By using certain devices such as alphabetical device and subject device. Now let us summarize the whole session. In this session, we described the dynamic theory of library classification formulated by Dr. S.R. Ranganathan and differentiated it from the descriptive theory. We have also studied the significance of normative principles in the dynamic theory. The different levels of normative principles are explained. Ranganathan recognized 3 planes of work in classification. An attempt is made to state and explain the canons for work in the idea plane. Here are few assignments for you to work out. First one, describe briefly the major characteristics of the dynamic theory of library classification developed by Dr. S.R. Ranganathan. Second, state and explain the canons for array with suitable examples. Third, examine how far the classification schemes DDC and CC achieves conformity to the canon of consistent sequence. Fourth, what do you mean by a coalesced array? Explain with an illustrative example how a coalesced array can achieve filiatory sequence among classes. Now I'll provide you with few books for further reading. First one, Prolegomena to Library Classification, 3rd edition, written by Dr. Ranganathan S.R., published by Sharda Ranganathan Endowment for Library Science, 1967. Second, Theory of Classification, 4th edition revised, written by Krishnan Kumar, published by Vikas Publishing House, New Delhi, 1988. Third, Library Classification, Facets and Analysis, written by Hussain Shahabat, published by Tata McGraw-Hill, New Delhi, 1993. Fourth, Bachelor of Library Science, Study Material, University of Calicut, 1995. Thank you for watching this session. See you in the next session. Until then, bye.
Welcome to Interface. Today we have with us an eminent scholar, writer, poet and a microbiologist with us as our guest. Ma'am, Mariam Shinasi, we would like to know your career as a microbiologist.
Well, when I decided to study biology, my focus was on microbiology, and then I enrolled in Faculty of Science, and I've been accepted in biology department, and then I carry on with it, focusing on microbiology because I do believe that I need to learn a lot about the microorganism, what is happening in that small environment, and what the interaction between different kind of microorganisms. And after that, I carried on with my PhD, and I left my country to go for about 5 years to Scotland to study microbiology and focusing on the aflatoxin and the carcinogenic things. It's produced from different kind of fungus. It is naturally a carcinogenic chemical. My thesis was focusing also on very specific fruits, as date fruits, which is available and valuable fruits in my country. And we would like to check if there is a possibility for this aflatoxin to produce in native fruits under the natural environment, or it depends to the normal conditions. So that was my PhD for about 4 years when I was studying in Glasgow Caledonian University.
Yeah, being a microbiologist, you contributed a lot to your country. At the same time, you turn into a publisher. But that's a very rare case. A woman, especially from the Arab traditional family, to become a publisher is a very laborious task. But you are very well established publisher there. What is your opinion about this field? Is it a very simple one or a very difficult one?
Mm-hmm. To shift to be a publisher and for publishing area, it didn't come suddenly, it came gradually, because I think what we got in my country with very good education, established education, especially in my generation, it was, I think we were the first generation who got different education, and we got a systematic education compared to the past, where it is very simple to be learn few things, like learning Arabic, the basic thing, learning Quran and some mathematics and all those things. But after 1971, when we established the country and we got establishing also the education, we have the base for the education. So it's encouraging us to improve our skills in different areas. One of the skills should be improved in writing. So I liked writing, so it was just coming gradually. But my focus was in science. Why in particular science? Because I do believe that it is in science. I need to learn it I need to learn laboratory, I need to study it, I need somebody to help me to discover all those things. But with publishing and writing, it is just skills. So here where I need to improve my skills. So once I graduate from the PhD, and then I published several books, several papers, and I contributed to many conferences. I published refereed paper. So it's about like 48 publishers was published in my subject. And after that I worked in the university as well for 5 years, University of Sharjah, and then I moved to Ministry of Environment and Water. So it's linked all with my career, but still I felt like I've got skills where it needs to be improved. So then I just moved. So it came gradually.
Yeah, this was quite natural growth.
Yeah, I think so. I do believe in that.
Yeah, that's all right. As a publisher and as a woman, what is the obstacles you faced?
Well, this is a question quite normal now in my life. I mean, if I faced any barriers, if there is any something happened or something, but I felt like it is my decision, where it cannot be stopped by anyone. If I've got skills and I do believe in it, so I need to improve it, and I need to spend some time to improve these skills. And once I publish in Arabic, in my language, because in science I need to publish most of the time in English, and it's all refereed papers in international journals, and sometimes in international conferences, based only on scientific solid base or scientific information. And it is laboratory work, which it should be transferred to paper, this journal paper or scientific paper. And writing, it is my skills, my experiences, and how I contribute this also with large people. I mean, it's not limited like scientific, it's only with scientists, but with writing novels, books in different areas, I can contribute this with different people. I mean, not only with specific or specialist people. So here it is guide me that it is, I need to know the world. I mean, I need to know whole people. I need to know more experiences. And this is what attracted me to India, because my father linked with India because he is a businessman, and he came to India, especially in Bombay. And this is more than 40 or 50 years. And he feel like India is part of his life, and we learned this from him, and he explained a lot about the people who he met and what he learned. And when even we received the guests from India, so they are very welcome to my house. So this is how we contribute with a great country like India. It's a rich country in different areas. So this is, I felt like it is no barriers, or it is, I feel like my life is still, I feel it like it's a basic. Some people, they feel it is amazing in part of it, but I feel it like it's worth to be contributed with others. This is also how I look to it.
In a way, you are trying to make a contribution as did in the past. As you know, many Arabs contributed a lot to translate the Indian works like Siddhanta and other things. In such case, you are filling the gap between the past and present. Actually, past there was a great tradition of translation, just like the Baitul Hikmah. After that, that continuum was stopped. But now, due to your Yasmin publication and others, Kalima publication, it has been restarted, revamped. Is there any plan to continue this one?
The people, how they like reading, education, they do believe in it, they respect the educated people, so that all encouraged me to come. And it was, I think it is in my storybooks or novel, it's a great thing that to translate directly from Arabic to Malayalam. We don't need some other language, international language.
The direct translation. Direct Arabic to Malayalam, or Arabic to Hindi, or Hindi to Arabic. Yes.
So this
there is no need of English as a linked language.
English or other language as well.
Yes.
So this is one of improving, I think, and it is a good step to start and to continue with it. And I think all of us, we do believe when the whole world contribute together, working together, we can exchange and learn. It depends what I need from this philosophy, Indian culture, Indian, it is rich civilization, and it has part of it is really very bright. And also in my culture, we need to exchange from each others. I mean, traveling, it is a really great thing when we travel abroad and see people, different culture, different mentality, different way of thinking. And then when they read our storybooks, it is our life also, our experiences. And this is when I talk about only novel and some cultural books, but also we need to look to the science, philosophy, and other things. Darul Yasmin doing maybe a small part now. It's playing a really very small part. I mean, to encourage that it is, we need to learn different languages. We need to learn about others. We need to learn from their experiences, and how we can contribute in all those and play a role in all that and improve our life, the standard of living. I think not only in health but also in social, not only in social but also in economy. So it is different thing. Translation, no doubt that it is. We need this, and we need to work a lot about it. Yeah.
Yeah. You are president of UAE Publishers, and normally it is an area mastered by the male, but at the same time now a woman is in the position of a president. How do you feel that?
I think in my country it is completely different, because women now dominate in this.
Yeah.
So before it was Sheikha Badur. Yeah. She was the first woman to be president in Emirates, and now she is playing a part in internationally. Yeah. And then I came but I felt that it is because it is just 2 months since I've been appointed to be in this position. It was by election. It is, I think, something great to help everyone, especially the writers.
Yeah.
And writers all the time complain from publishers, and publishers thinking that they are victim of writers. So this relationship is still, it's not balanced still. Yeah, it is not balanced. And in my country also, we now trying to modify the vision of the publishing, what we need, what is the vision, what we need to do, and how it should. And it is modified in one sentence, maybe 2 words, that we need to improve should be industrial of publishing, because we need to respect the writer and the publisher in the same time, and they are equal. We cannot sacrifice one of them. We need both of them.
To balance them.
Yes, and to also balance the relationship between writer,
both publishers and writer.
But I feel like I'm more with the side of publishers, because I need to defend also their rights. Most of the time we're talking about the rights of the writers. And then we need also to talk about the rights of publishers. So a publisher put a lot of money, a lot of effort. They do the distribution, they travel, they try to announce, they try to advertise. They also play a role in it. So they are equal, and it should be balanced. If we want this to be moved to industrial, we say we have an Arab world or a country like United Arab Emirates, we need to work on it. So this will be balance between writers and publishers as well.
Yeah, it is known that you are a part and parcel of a Sadhana Cultural Foundation. I would like to know something about the Sadhana Cultural Foundation and its activities.
Yeah, Sadhana, it's because they heard about the Arabic festival, which I try to contribute with Calicut University. It is a historic university, and they rooted back to maybe years in Arabic culture, and the Department of Arabic it's also it's improved, and they put effort to give the best education for the student. So by having and hosting this festival, people learn a lot from the novel, from the writing, from what is going on, in writing, especially in Arabic countries. And they heard about this. They would like to participate and contribute in it. So we say this is your chance to also put a light on what you are doing in Sadhana. As the owner, Sheikha Asma bint Sagr al-Qasimi, Her Highness, she is supporting many writers. She is supporting any kind of cultural activities, and she was really happy to hear that it is in India. There are many people, they learn this about the Arabic art and poet, novel. They would like to read more, and also they like to read updated books.
To propagate Arab culture and heritage.
Yes, yeah.
It is well known fact that Sharjah is the cultural capital of UAE. And what is the role played by the Sharjah to disseminate the knowledge, to transparent the culture of the Arab nations?
I think I have to admit that it is all what we achieved in Sharjah city. It's belonging to His Highness Sheikh Sultan bin Mohammed al-Qasimi, the ruler of the Sharjah, and he put the pillars about cultural, and he put the pillars and seeds about what is culture, what is the book fair, the meaning of that thing, and he put many prizes, and he support the society by even giving us his experience in that. And I think this now we worked in this 40 years, and he did a lot, not only in publication and books and writing, also in theater, cinema, and translation. His book translated to many different languages, Indian language I'm talking in India. It is all true stories coming from the history, but it is maybe unknown history or part of the history. It is small stories happened in the past, and he is highlighting on those stories. So from him we learn a lot, especially in a school. They take us in different activities, and then gradually we learn and how the important about culture and what is the meaning of reading, and how this reading is enlightening our life. It is make a path for us, reading, whether I agree with you in the concept or I disagree, but at the end of the day we have different way of living, we have different way of things to do and to contribute or to discuss. For that, I think the main thing in Sharjah that it is should be admitted to His Highness Sheikh Sultan bin Muhammad Al-Qasimi.
Thank you, ma'am. Dr. Mariam Shinasi.
Hello everyone, welcome to the session on genome databases. In this session, we will study about the various kinds of genome databases, their organization and their attributes as well. We would also gain an insight about how to use information retrieval system to obtain data and the need to analyze sequence data. Introduction to biological databases. All living organisms are characterized by the capacity to reproduce and evolve. The genome of an organism is defined as the collection of data within that organism, including the set of genes that encode proteins. In 1995, the complete genome of a free living organism was sequenced for the first time, the bacterium Haemophilus influenza. In the few years since then, the genomes of thousands of organisms have been completely sequenced, ushering in a new era of biological data acquisition and information accessibility. Publicly available data banks now contain billions of nucleotides of DNA sequence data collected from over 260,000 different organisms. 3 publicly accessible databases store large amounts of nucleotide and protein sequence data. GenBank at the National Center for Biotechnology Information (NCBI) of the National Institute of Health (NIH) in Bethesda, the DNA database of Japan at the National Institute of Genetics in Mishima, and the European Molecular Biology Laboratory (EMBL) nucleotide sequence database at the European Bioinformatics Institute (EBI) in Hingston, England. These 3 databases share their sequence data daily. They are coordinated by the International Nucleotide Sequence Database Collaboration (INSDC). GenBank. GenBank is the NIH genetic sequence database, an annotated collection of all publicly available DNA sequences. GenBank is part of the International Nucleotide Sequence Database Collaboration, which comprises the DNA Databank of Japan (DDBJ), the European Molecular Biology Laboratory (EMBL), and GenBank at NCBI. These 3 organizations exchange data on a daily basis. A GenBank release occurs every 2 months and is available from the FTP site. The release notes for the current version of GenBank provide detailed information about the release and notifications of upcoming changes to GenBank. In more than 30 years since its establishment, GenBank has become the most important and most influential database for research in almost all biological fields, whose data are accessed and cited by millions of researchers around the world. GenBank continues to grow at an exponential rate, doubling every 18 months. Release 194, produced in February 2013, contained over 150 billion nucleotide bases in more than 162 million sequences. GenBank is built by direct submissions from individual laboratories as well as from bulk submissions from large-scale sequencing centers. Organisms in GenBank. Over 260,000 different species are represented in GenBank, with over 1,000 new species added per month. The number of organisms represented in GenBank is shown in the table. Eukaryotes have a nucleus and are often multicellular, whereas bacteria do not have a nucleus. Archaea are single-celled organisms distinct from eukaryotes and bacteria, which constitute a third major branch of life. Viruses, which contain nucleic acids, DNA or RNA, but can only replicate in a host cell, exist at the borderline of the definition of living organisms. From the table, we can see that the organisms in GenBank constitute mostly of eukaryotes. Of the microbes, there are currently over 25 times more bacteria than archaea represented in GenBank. To help organize the available information, each sequence name in a GenBank record is followed by its data file division and primary accession number. The following codes are used to designate the data file divisions: PRI, which denotes for primate sequences, ROD for rodent sequences, MAM for other mammalian sequences, VRT other vertebrate sequences, INV invertebrate sequences, PLN plant, fungal and algal sequences, BCT bacterial sequences, VRL viral sequences, PHG bacteriophage sequences, SYN synthetic sequences, UNA unannotated sequences, EST sequences, PAT patent sequences, STS sequences or sequence tag sites, GSS sequences or genome survey sequences, HTG high throughput genomic sequences, HTC high throughput cDNA sequences, and ENV environmental sampling sequences. Types of data in GenBank. There are a number of molecular sequences in GenBank. A fundamental distinction is that DNA, RNA based and protein sequences are stored in discrete databases. Furthermore, within each database, sequence data are represented in a variety of forms. For example, beta globin may be described at the DNA level, for example as a gene, at the RNA level as a messenger RNA or mRNA, at the protein level. Because RNA is relatively unstable, it is typically converted to complementary DNA or cDNA, and a variety of databases contain cDNA sequences corresponding to RNA transcripts. Thus, for our example of beta globin, the various forms of sequence data include the following: genomic DNA databases. Beta globin is part of a chromosome. In the case of human RBP, we will see that its gene is situated on chromosome 11. Beta globin may be a part of a large fragment of DNA such as a cosmid, bacterial artificial chromosome or BAC, yeast artificial chromosome or YAC that contain several genes. A BAC is a large segment of DNA, typically about 200,000 base pairs or 200 kilobases, that is cloned into bacteria. Similarly, YACs are used to clone large amounts of DNA into yeast. BACs and YACs are useful vectors with which to sequence large portions of genomes. Beta globin is present in databases as a gene. The gene is the functional unit of heredity. It is a DNA sequence that typically consists of regulatory regions, protein coding exons and introns. Often, human genes are 10 to 100 kb in size. Beta globin is present as a sequence tagged site, STS, that is, as a small fragment of DNA, typically 500 base pairs long, that is used to link genetic and physical maps, and which is part of a database of sequence tagged sites or DBSTS. cDNA databases corresponding to expressed genes. Beta globin is represented in databases as an expressed sequence tag or EST, that is, a cDNA sequence derived from a particular cDNA library. If one obtains a tissue such as liver, purifies RNA, then converts the RNA to the most stable form of cDNA, some of the cDNA clones contained in that cDNA are likely to encode beta globin. Expressed sequence tags or ESTs. The database of expressed sequence tags DBEST is a division of GenBank that contains sequence data and other information on single pass cDNA sequences from a number of organisms. An EST is a partial DNA sequence of a cDNA clone. All cDNA clones and thus all ESTs are derived from some specific RNA source such as human brain or rat liver. The RNA is converted into a more stable form cDNA, which may then be packaged into a cDNA library. ESTs are typically randomly selected cDNA clones that are sequenced on one strand and thus may have a relatively high sequencing error rate. ESTs are often 300 to 800 base pair in length. The earliest efforts to sequence ESTs resulted in the identification of many hundreds of genes that were novel at the time. In November 2008, GenBank had over 5 crores 80 lakhs ESTs. Currently, GenBank divides ESTs into 3 major categories: human, mouse and other. The table shows the 10 organisms from which the greatest number of ESTs has been sequenced. Assuming that there are 22,000 human genes and given that there are about 8.1 million human ESTs, there is currently an average of over 300 ESTs corresponding to each human gene. ESTs and Unigene. The goal of the Unigene or Unique Gene project is to create gene-oriented clusters by automatically partitioning ESTs into non-redundant sets. Ultimately, there should be one Unigene cluster assigned to each gene of an organism. There may be as few as one EST in a cluster, reflecting a gene that is rarely expressed, to tens of thousands of ESTs associated with a highly expressed gene. There are over 100 organisms currently represented in Unigene. For human beta globin, there is only a single Unigene entry. This entry currently has 2,400 human ESTs that match the beta globin gene. This large number of ESTs reflects how abundantly the beta globin gene has been expressed in cDNA libraries that have been sequenced. A Unigene cluster is a database entry for a gene containing a group of corresponding ESTs. There are now thought to be approximately 22,000 human genes. One might expect an equal number of Unigene clusters. However, in practice there are more Unigene clusters than there are genes. Currently, there are about 120,000 human Unigene clusters. This figure is a schematic representation of Unigene clusters. ESTs are mapped to a particular gene and to each other. The number of ESTs that constitute a Unigene cluster varies from 1 to tens of thousands. On an average, there are 300 human ESTs per cluster. Genome databases at EBI. The European Bioinformatics Institute or EBI is an EMBL outstation located at Hingston Hall near Cambridge, UK. Since September 1994, all activities previously based at the EMBL data library in Heidelberg, Germany are located at the EBI. The database services of the EBI are the continuation and extension of EMBL data library. A central activity of the European Bioinformatics Institute (EBI) is the development and distribution of the EMBL nucleotide sequence database. The EBI also maintains and distributes the Swiss prot protein sequence database in collaboration with Amos Bayroch of the University of Geneva. Over 50 additional specialist molecular biology databases, some produced in collaboration with the EBI, are also distributed through EBI releases and network services. The first completed genomes from viruses, phages and organelles were deposited into the EMBL database in the early 1980s. Since then, molecular biology shift to obtain the complete sequence of as many genomes as possible, combined with major developments in sequencing technology, resulted in hundreds of complete genome sequences being added to the database, including archaea, bacteria and eukaryota. The EBI website is comparable to NCBI in its scope and mission, and it represents a complementary independent resource. EBI features 6 crore molecular databases, which are as follows. Number 1, EMBL Bank is the repository of DNA and RNA sequences that is complementary to GenBank and DDBJ. Number 2, SwissProt. Number 3, TREMBL are 2 protein databases. Number 4, MSD is a protein structure database. Number 5, Ensemble is one of the 3 main genome browsers. Number 6, ArrayExpress is one of the 2 main worldwide repositories for gene expression data, along with the Gene Expression Omnibus at NCBI. Genome analysis of model organisms. A major application of bioinformatics is the analysis of the full genomes of organisms that have been sequenced starting in the late 1990s, including microbial genomes, the budding yeast Saccharomyces cerevisiae, the nematode worm Caenorhabditis elegans, the plant Arabidopsis thaliana, the fruit fly Drosophila and the human genome. Many additional genome sequencing projects are either being planned or are already underway. Many years of genetic and biochemical research of these model organisms, the bacterium Escherichia coli, Saccharomyces cerevisiae, Caenorhabditis elegans, Arabidopsis thaliana and Drosophila melanogaster have led to the accumulation of a large amount of information on gene organization and function. The mouse Mus musculus is a genetic model for humans because the 2 species are so closely related through evolution. A newly identified gene in another organism can be compared to the existing database of information to find whether it has a similar function. Genes involved in human disease, for example, are sometimes found to be similar to a fruit fly gene at the protein sequence level. As a result, a similar gene or family of genes may be found in several organisms, including a model organism, but the function is not known because the gene functions have not yet been analyzed. Hence, continued biological analysis of model organisms in those areas that are not tractable by the tools of bioinformatics has many important applications. Viral genomics and bioinformatics. From the recognition by Ivanovsky in 1892 that tobacco mosaic disease is caused and transmitted by fine pore filtrates, viruses have been isolated, characterized, identified and studied from animals, plants, protists, bacteria and even other viruses. As human and global public health pathogens that can be highly contagious and have devastating morbidity and mortality consequences, viruses are the focus of much research. The difficult challenge has been to define and study a minuscule being with the appropriate tools. In the past, these tools often provided only low resolution views. A first approach to studying an unknown virus is to know exactly its identity and to place it into context of other related and non-related viruses. For human and public health, this is important as the identity may provide a course of action to limit the effects of the pathogen. Although in general, large data sets of related viral genomes from a family are just being assembled, there are available sets of genomes from members of certain virus families that are of recent and critical interest in the context of human and public health issues. Many of these data sets have either been recently assembled and reported, such as rhinovirus, or have been reviewed recently, for example rhabdoviruses, papillomavirus and human immunodeficiency virus or HIV. Rather than attempting to provide a comprehensive survey of many virus families with multiple genomes sequenced and analyzed, A goal of this special issue is to provide a sampling and a range of the virus families that have been and are of importance, such that multiple genomes have been sequenced and analyzed using bioinformatics methods. From these contributions, one will see that genomics and bioinformatics are intertwined. At one end of the spectrum are human adenovirus, which contain DNA genomes of 35 kb. Once considered large genome viruses, these are provided in contrast to the much larger genomes found amongst the Iridoviridae with DNA genomes of 106 kb. Eaton et al. Reviews these. In addition, Hendrickson et al. Examines the Poxviridae genomes, which range from 130 to 375 kb. For comparison, Wu et al. Reports on the coronaviruses with the largest genomes found for RNA viruses, that is 26 to 38 kb. This report on coronavirus illustrates the importance of viral genomes and bioinformatics in monitoring and identifying global pathogens rapidly and effectively, allowing for measures to be taken. The NCBI database provides a comprehensive list of 6021 viral genomes, their features, accession number, host, and the taxonomic details. Genomics of bacteria and archaea. The first bacterial genome was sequenced in 1995 and the first archaeal genome in 1996. Soon after these breakthroughs, an exponential rate of genome sequencing was established, with a doubling time of approximately 20 months for bacteria and approximately 34 months for archaea. Comparative analysis of the hundreds of sequenced bacterial and dozens of archaeal genomes leads to several generalizations on the principles of genome organization and evolution. A crucial finding that enables functional characterization of the sequenced genomes and evolutionary reconstruction is that the majority of archaeal and bacterial genes have conserved orthologs in other, often distant organisms. This is a screenshot of UCSC Archaea genome browser. Despite the tremendous variety of lifestyles as well as metabolic and genomic complexity, bacterial and archaeal genomes show easily discernible common architectural principles. The sequenced bacterial genomes spans 2 orders of magnitude in size from 180 kb in the intracellular symbiont Carsonella rudi to 13 mb in the soil bacterium Sorangium cellulosum. Remarkably, bacteria show a clear-cut bimodal distribution of genome sizes, with the highest at 2 mb and the second smaller one at 5 mb. Although there are many genomes of intermediate size, this distribution suggests the existence of 2 more or less distinct classes of bacteria. The possibility remains that the bimodality of the bacterial genome size distribution is due to the bias of the genome sequencing efforts towards smaller genomes, such as those of symbionts and parasites. But with the growth of the genome collection, this explanation is becoming increasingly less plausible. Archaea are less diverse in genome size, from around 0.5 mb in the parasite Nanoarchaeum equitans to approximately 5.5 mb in Methanosarcina barkeri, and show a sharp peak at approximately 2 mb that almost precisely coincides with the position of the highest bacterial peak, and a heavy tail corresponds to larger genomes. As the representation of archaeal genomes in the current database is much less complete than the representation of bacterial genomes, it remains to be seen whether the genome size distributions in archaea and bacteria are genuinely different, or the differences only reflect sequencing biases. That is, a second peak might appear in the archaeal distribution once additional larger genomes of mesophilic archaea are sequenced. All very small, approximately 1 mb, with a current record belonging to the abundant marine, a proteobacterium Pelagibacter ubique SAR11 at 1.3 mb. Notably, with the progress in genomics, it has become clear that there is no gulf in the genome sizes between bacteria and archaea, viruses and eukaryotes. Genomes of model organisms. A model organism is a non-human species that is extensively studied to understand particular biological phenomena, with the expectation that discoveries made in the organism model will provide insight into the workings of other organisms. Model organisms are in vivo models and are widely used to research human disease when human experimentation would be unfeasible or unethical. This strategy is made possible by the common descent of all living organisms and the conservation of metabolic and developmental pathways and genetic material over the course of evolution. Studying model organisms can be informative, but care must be taken when extrapolating from one organism to another. In researching human disease, model organisms allow for better understanding the disease process without the added risk of harming an actual human. The species chosen will usually meet a determined taxonomic equivalency to humans so as to react to disease or its treatment in a way that resembles human physiology as needed. Although biological activity in a model organism does not ensure an effect in humans, many drugs, treatments and cures for human diseases are developed in part with the guidance of animal models. There are three main types of disease models: homologous, isomorphic and predictive. Homologous animals have the same causes, symptoms and treatment options as would humans who have the same disease. Isomorphic animals share the same symptoms and treatments. Predictive models are similar to particular human disease in only a couple of aspects, but are useful in isolating and making predictions about the mechanism of a set of disease features. Model organisms are chosen on the basis that they are amenable to experimental manipulation. This usually will include characteristics such as short life cycle, techniques for genetic manipulation such as inbred strains, stem cell lines and methods of transformation, and non-specialist living requirements. Sometimes the genome arrangement facilitates the sequencing of the model organism's genome, for example by being very compact or having a low proportion of junk DNA, for example yeast, Arabidopsis or pufferfish. When researchers look for an organism to use in their studies, they look for several traits. Among these are size, generation time, accessibility, manipulation, genetics, conservation of mechanisms and potential economic benefit. As comparative molecular biology has become more common, some researchers have sought model organisms from a wide assortment of lineages on the tree of life. Important model organisms. Model organisms are drawn from all three domains of life as well as viruses. The most widely studied prokaryotic model organism is Escherichia coli or E. coli, which has been intensively investigated for over 60 years. It is a common gram-negative gut bacterium which can be grown and cultured easily and inexpensively in a laboratory setting. It is the most widely used organism in molecular genetics and is an important species in the field of biotechnology and microbiology, where it has served as the host organism for the majority of work with recombinant DNA. Simple model eukaryotes include Baker's yeast such as Saccharomyces cerevisiae and fission yeast such as Saccharomyces pombe, both of which share many characters with higher cells, including those of humans. For instance, many cell division genes that are critical for the development of cancer have been discovered in yeast. Chlamydomonas reinhardti, a unicellular green algae with well-studied genetics, is used to study photosynthesis and motility. C. reinhardti has many known and mapped mutants and expressed sequence tags, and there are advanced methods for genetic transformation and selection of genes. Dictyostelium discoideum is used in molecular biology and genetics and is studied as an example of cell communication, differentiation and programmed cell death. Among invertebrates, the fruit fly Drosophila melanogaster is famous as a subject of genetics experiments by Thomas Hunt Morgan and others. They are easily raised in the lab with rapid generations, high fecundity, few chromosomes and easily induced observable mutations. The nematode Caenorhabditis elegans is used for understanding the genetic control of development and physiology. It was first proposed as a model for neuronal development by Sidney Brenner in 1963 and has been extensively used in many different contexts since then. Caenorhabditis elegans was the first multicellular organism whose genome was completely sequenced and as of 2012 the only organisms to have its connectome or neuronal wiring diagram completed. Arabidopsis thaliana is currently the most popular model plant. Its small stature and short generation time facilitates rapid genetic studies and many phenotypic and biochemical mutants have been mapped. Arabidopsis was the first plant to have its genome sequenced. Among vertebrates, guinea pigs, Cavia porcellus were used by Robert Koch and other early bacteriologists as a host for bacterial infections, becoming a byword for laboratory animal, but are less commonly used today. The classic model vertebrate is currently the mouse, Mus musculus. Many inbred strains exist as well as lines selected for particular traits, often of medical interest, for example body size, obesity, muscularity and voluntary wheel running behavior. Among vertebrates, the rat, Rattus norvegicus, is particularly useful as a toxicology model and as a neurological model and source of primary cell cultures owing to the larger size of organs and suborganellar structures relative to the mouse, while eggs and embryos from Xenopus tropicalis and Xenopus levis or the African clawed frog are used in developmental biology, cell biology, toxicology and neuroscience. Likewise, the zebrafish Danio rario has a nearly transparent body during early development, which provides unique visual access to the animal's internal anatomy during this time period. Zebrafish are used to study development, toxicology and toxipathology, specific gene functions and roles of signaling pathways. Other important model organisms and some of their uses include T4 phage or viral infection, Tetrahymena thermophila for intracellular processes, maize for studying transposons, hydras for regeneration and morphogenesis, cats for neurophysiology, chickens for development, dogs for respiratory and cardiovascular systems, Northobranchius furzeri for aging and non-human primates such as the rhesus macaque and chimpanzee for hepatitis, HIV, Parkinson's disease, cognition and vaccines. Ensemble is a joint scientific project between the European Bioinformatics Institute and the Wellcome Trust Sanger Institute, which was launched in 1999 in response to the imminent completion of the Human Genome Project. After 10 years in existence, Ensemble's aim remains to provide a centralized resource for geneticists, molecular biologists and other researchers studying the genomes of our own species and other vertebrates and model organisms. Ensemble is one of the several well-known genome browsers for the retrieval of genomic information. Similar databases and browsers are found at NCBI and the University of California, Santa Cruz, UCSC. This is a screenshot of Ensemble database. UCSC Genome Browser. The UCSC Genome Browser is an online genome browser hosted by the University of California, Santa Cruz, UCSC. It is an interactive website offering access to genome sequence data from a variety of vertebrate and invertebrate species and major model organisms, integrated with a large collection of aligned annotations. The browser is a graphical viewer optimized to support fast interactive performance and is an open source web-based tool suite built on top of a MySQL database for rapid visualization, examination and querying of the data at many levels. The Genome Browser database browsing tools, downloadable data files and documentation can all be found on the UCSC Genome Bioinformatics website. This is a screenshot of the UCSC Genome browser. Let us now summarize what we have discussed so far. Genome database collect genome sequences, annotate and analyze them, and provide public access. Some add curation of experimental literature to improve computed annotations. These databases may hold many species, genomes, or a single model organism genome. Viruses have been known and studied since before the start of the last century. Continued investigations of these beings over these many years, despite the lack of high-resolution tools, reflect the many important roles they played relative to humans. Our understanding of them and our ability to take advantage of them are limited by our lack of complete data, preferably high resolution, and by the tools that are available. The relevant and important contributions in this special issue, along with the advent of recombinant DNA technology and DNA sequencing methodology near the turn of this century, and with the continuing development of the next generation DNA sequencing technology, highlight how genomics and bioinformatics are allowing and will allow a much deeper and broader understanding and appreciation of viruses, bacteria, bacteriophages, and archaea. Here are a few questions for you to work out. Number 1, what are genome databases? Give a few examples. Number 2, what are the main components of a genome database? Number 3, what are model organisms? Explain how they are used in biological research. Number 4, explain how bioinformatics has helped in studying about viral genomes. Number 5, what are the applications of studying bacterial genomics? Here are a few books for your further studies. Bioinformatics, Concepts, Skills and Applications, written by S.C. Rastogi, Namita Mendirita, Parag Rastogi, published by CBS Publishers and Distributors, 1993, first edition. Understanding Bioinformatics, written by Marketa Swellable, Jeremy Obaum, published by Garland Science Publishers, 2008. Bioinformatics, Databases, Tools and Algorithms, written by Orpita Basu, Siminder Kaur Tukral, published by Oxford University Press, 2007. Thank you for watching this program. Here we conclude the session on genome databases. I hope this session was very useful for you. Goodbye.
Hello and welcome to a session on fundamentals of program learning. If we look at the history of education instruction, we can see that the method of mass instruction has been traditionally used in schools. In this method, the content, the instructional technology and learning materials are same for all students in a classroom. Moreover, all students are expected to keep the same pace of learning. As we know, a class is comprised of individual students who differ from one another in cognitive capacities, skills, perceptions, academic performance and learning experiences. The pace of learning also varies from one student to another. Mass instruction method does not take into consideration such differences and unique requirements of individual students. In order to rectify this problem, a new method of instruction was introduced. This new method is called individualized instruction, which takes into account the learning abilities and interest of individual learners. As the name suggests,
Individualized instruction places emphasis on the learning experiences of individual students, not on class as a whole. The primary goal of this method of instruction is to make learning self-directed and self-initiated. We have seen that educational technology plays an important role in individualizing instruction. In the past few decades, several new techniques have been introduced with the aim of making individualized instruction more effective. In this unit, we will learn more about one of those techniques used in individualized instruction, namely program learning. What is program learning? Program learning, also known as programmed instruction, is one of the most effective innovations introduced to the teaching learning process. It is a highly individualized and systematic strategy of instruction. It is useful not just in classroom instruction but also in self-learning and auto instruction. To put simply, program learning is the instruction provided by means of program learning materials. The program used in this method is designed and sequenced, anticipating the responses of students in the teaching learning process. Thus, the entire range of learning experience is pre-designed based on a set of predetermined instructional goals and objectives. Let us consider an example to understand the concept of program learning better. Many Indian universities are providing correspondence courses on a wide range of subjects. The students enrolled in these courses do not or rarely interact with regular tutors or instructors. Instruction, on the other hand, is provided to these students through carefully designed learning material which helps them in the process of self-instruction. It is important here to note that the scope of program learning is not limited to self-instruction courses. It is widely used in regular class settings as well. Program learning applies the principles of psychology and technology to provide individualized instruction to students and improve the effectiveness and quality of learning process. Key terms used in the program learning. Before we examine this method of instruction in detail, it is important to clarify some of the terms we constantly use in relation to program learning. Program. The term program refers to the subject matter to be learned by the students. It is an instructional sequence comprised of several small units of information. A program is designed based on instructional objectives, and the aim of a program is to help students achieve these objectives through teaching learning process. Frames. As already mentioned, a program contains several units of information. In other words, learning material is presented to students in the form of small units which are sequentially arranged. These small units are called frames. It should be noted here that these frames are graded based on their level of difficulty and presented to students stage by stage. Sequence. The term sequence refers to the order in which frames are arranged in a program and presented to students during the process of instruction. Programming. In the context of program learning, the term programming refers to the process of arranging the subject matter to facilitate effective learning. Programmer. The teacher or instructor who develops the program which are to be used in programmed instruction is called a programmer. It is important to note that a programmer should be subject matter expert with adequate knowledge in technology and psychology. He or she should be familiar with the technique of programming too. Definitions, characteristics and principles of program learning. Many scholars trace the origin of program learning to the great Greek philosopher Socrates, who is believed to have developed a program in geometry. However, this instruction method, as we understand it today, was developed in the 20th century by a number of American psychologists including Edward Thorndike, Sidney L. Presley, B.F. Skinner, G. Holland and Norman A. Crowder. Of these, the names of Skinner and Crowder deserve a special mention since they are considered as developers of linear programming and branched programming respectively. Definitions of program learning. Different scholars have defined program learning in different ways. These definitions throw light on the chief characteristics of this method. Let us go through some of the most popular definitions of program learning. Susan Murphy defined program learning as the method of designing a reproducible sequence of instructional events to produce a measurable and consistent effect on the behavior of each and every acceptable student. According to Harold D. Bernard, programmed learning refers to the arrangement of instructional material in progressive sequences. Wendell I. Smith and J. William Moore defined programmed instruction as the process of arranging the material to be learned into a series of sequential steps. They further identified how program learning functions. According to them, it usually moves students from a familiar background into a complex and new set of concepts, principles, and understanding. Main characteristics of program learning. Program learning has a set of defining characteristics that differentiate it from other methods of instruction. Here are some of the most important characteristics of this method. To begin with, it is a method of individualized instruction where learning material and information are presented to students to suit their individual needs. This helps to maximize the rate and depth of learning, fosters understanding and motivates students. Another important characteristic of this method is self-pacing. This means that students learn and move forward at their own speed and pace. It acknowledges the fact that there are differences in the rate at which individuals learn. The subject matter is broken into smaller units and presented to students in a sequential order. By presenting the material through small units and in a progressive manner, the level of complexity is increased gradually from lower to the higher levels. It also ensures smoothness of transition from one item to another and eliminates the possibility of errors. In this method, interaction happens between students and the program. Needless to say, it requires students to remain active throughout the process of instruction. Program learning ensures continuous responses from students. In this method, it is possible to immediately confirm whether the responses and answers given to students are correct or not. It also gives students a chance to correct their wrong responses before they move on to the next stage of learning. In other words, at each step, students know how they are doing and can assess their own performance. Another important characteristic of this method is the organized nature of the knowledge. Concepts and information are organized in a progressive manner, starting from the least complex to the most complex. This ensures continuity between easier and difficult concepts. It also ensures that students have mastered the basic and easier concepts before more complex and difficult ones are presented to them. The instructional sequence of program learning take into account the initial behavior of students. The program is designed keeping in mind the terminal competency students are expected to achieve at the end of the instructional process. Program learning ensures constant evaluation by recording the responses of students at each step. The performance and the progress made by students can be evaluated by checking the nature of the responses. The students' responses can also be used to improve the overall quality of the program. Program learning offers individualized instruction by means of a variety of sources such as program textbooks, teaching machines, or computers. Instruction can be provided with or without the aid of teachers. Principles of program learning. A good program learning strategy is designed according to a set of fundamental principles. They are the principle of smaller steps. We have already seen that in program learning, the learning material is presented to students in smaller units which are arranged in a progressive sequence. The rationale behind this assumption is that students can learn better if the contents of a subject matter is presented to them in proper small steps. The principle of active response. Another underlying principle of this method is that students can learn better when they remain active in the process of instruction. This means that students should respond actively to every frame presented to them. When designing programmed material, proper care should be taken to involve students in each and every step of the learning process. The principle of immediate reinforcement. The third guiding principle of program learning is the psychological phenomena of reinforcement. This is based on the assumption that students learn better and feel motivated when they receive immediate feedback about the responses. Thus, a good program should make provisions to give immediate and constant reinforcement to students. The principle of self-pacing. As already mentioned, program learning is a method of individualized instruction. Therefore, an important principle of this method is self-pacing. That is, students learn better when they are allowed to learn at their own pace. A good program should be designed taking into account the individual differences of students. The frames should be presented in such a way that students are able to respond and move from one frame to another at their own speed and pace. The principle of continuous evaluation. Continuous evaluation helps to improve the quality of teaching as well as learning. In program learning, adequate care should be given to obtain and record student responses at every stage. This not only helps to evaluate the progress and made by students, but also to assess the overall effectiveness of the program. Program development. Development of program is a highly dynamic, extremely challenging and time-consuming task. Typically, it involves 3 stages, namely the preparation stage, construction or writing stage, and evaluation stage. Each stage consists of a series of steps and activities which a programmer has to carry out. Let us examine these stages and steps in some detail. 1. Preparation stage. Preparation is the first stage of program development. It is usually done in different steps, starting from the selection of a unit to the preparation of the content outline. Selecting a unit. As the first step of preparation, the programmer should choose a unit that is to be programmed. It is desirable that the programmer chooses a unit from his or her field of study. An important criterion for choosing the unit is the ease of handling learning material. The length of the unit should be determined based on the predefined instructional objectives. It is also important to choose short units instead of choosing the entire course content. Special care should be taken to choose units which are usually considered as a stumbling block to students. Another important criterion is to the logical order of the material. Special needs of students should also be given adequate consideration. Writing assumptions about students. The nature and content of the program differ for different target groups. Hence, it is important to list out the characteristics of the group for whom the program is being developed. That is to say, the programmer should clearly, objectively, and accurately write down all assumptions about the target group. This includes their age, skills, interest, background, etc. Teachers can refer to the cumulative records, case history, records of achievement test, aptitude test, and their personal experience to gather a clear picture of the target group. Determining instructional objectives. The instructional objectives should be determined prior to the development of the program. Moreover, these objectives should be stated in behavioral terms. Decisions regarding subject matter, instructional methods and learning material depend upon the instructional objectives. Defining prerequisite skills and knowledge. Any learning program is developed based on the assumption that the target group already possesses certain skills and knowledge. These prerequisite skills and knowledge should be stated in behavioral terms. This will help the programmer to demarcate the initial behavior with which students enter the learning process and the terminal behavior which they are expected to acquire. Preparing a criterion test. The next step in the preparation stage involves the construction of a criterion test. The purpose of this test is to show the range of the terminal behavior which is to be developed through the program. Developing content outline. This is the last step in the preparation stage. The programmer should write a clear and specific outline of the content that is to be programmed. Along with the outline, the order in which the learning material is to be presented should also be decided. 2. Construction or writing stage. This stage involves the construction and actual writing of the program. Here, programmers use the information they have gathered during the preparatory stage. A program can be written in different styles such as linear style or branched style. In the construction stage, the programmer takes decisions regarding which style is to be used. It is important to note that writing a programmed learning material differs from writing a textbook. The material should be prepared in line with the principles of program learning. The material is to be broken into small frames which are logically sequenced. Special care should be taken to keep the students meaningfully active by inviting responses at each stage. The frames should be designed in such a way that students get immediate feedback and reinforcement as they proceed from one frame to another. The task of writing the program is typically carried out in 3 steps, namely designing the frames, sequencing the frames and editing the program. The first step involves the designing of frames. Here the programmer should use special techniques like priming and prompting to help students respond correctly to the questions and proceed successfully from one frame to another. The second step involves proper sequencing of the frames. Here the programmer has to arrange the frames systematically in a logical order. Proper sequencing of frames leads students from their entry behavior to the terminal behavior. The frames are sequenced and ordered using logical and psychological principles. The third step of program writing is editing. Once the first draft of the program is written, the programmer should subject the draft to thorough review and editing to eliminate all possible errors. Technical inaccuracies, language inaccuracies and program inaccuracies should be removed completely before the final program is presented to students. 3. Evaluation stage. This is the third and final stage of the program development. In this stage, the programmer attempts to test the efficiency and effectiveness of the program he or she has developed. Evaluation is done in the light of the results obtained from testing. Moreover, modifications and revisions are also made to improve the quality of the program. Before the program is actually presented to the target group, the programmer may choose to conduct tryout testing on a sample of individual students or small group of students. Module 4. Traditional instructions versus program learning. Program learning is different from the traditional method of classroom instruction in many aspects. We have briefly touched upon some of those differences in the last 3 units. We can summarize the major differences between these 2 methods as follows. The traditional method involves mass instruction, while program learning involves individualized instruction. In the traditional method, the subject matter is presented to students as whole, but program learning presents the subject matter in small frames and step by step. In the traditional method, students do not get immediate feedback and reinforcement, but program learning offers immediate feedback and reinforcement to students. In the traditional method, the instructional objectives might not be well defined or they are defined vaguely. Program learning, on the other hand, states the objectives clearly in operational and behavioral terms. Program learning differs from the traditional method in the degree of preparation and precision. While the traditional method involves lesser degrees of preparation on the part of the instructor, programmers prepare learning materials with precision and care. In the traditional method, the role of students is minimal and limited to that of passive listeners. They remain passive during the instructional process while instructors do the summarizing and reviewing themselves. In program learning, students play an active role as the frames are designed in such a way that students naturally participate in the process by making constant responses. The unit of information presented to students is relatively lengthy in the traditional method, while program learning uses smaller units of information. In program learning, it is easy to modify the frames and sequences of frames on the basis of student reactions. Effective sequences of frames are retained and ineffective sequences of frames are discarded or modified through a series of tryouts and testing. However, such a gradual modification and revision is very difficult to implement in the traditional method. Advantages and limitations of program learning. Advantages. The greatest advantage of program learning is that it individualizes the process of instruction, giving students a chance to learn material at their own pace and speed. This method of instruction is an excellent solution to the problem of individual differences, which traditional method often fail to address. This method helps to keep students active and alert throughout the process of instruction. Better quality of learning experience is ensured through making students active participants in the process of instruction. The structure and design of the frames used in program learning make it possible to teach complex subject matters in an easy and systematic manner. Moreover, teachers can use this method to supplement the study of regular textbooks. Also, immediate feedback and reinforcement promotes effective learning. The program designed in this method can be used in self-instructional devices such as computer, teaching machine, program text, etc. These devices not only promote self-instruction but also solve the problem of the shortage of qualified instructors. Program learning is useful in the enrichment of curriculum and can be used to provide guidance and remedial instruction to students. Limitations. Despite all the above mentioned advantages, program learning has some serious limitations. Though this method individualizes instruction, it is not as interactive as a classroom discussion or computer-assisted instruction. Students cannot alter the scope or sequence of learning which are predefined by the programmer. Thus, this method limits students' freedom of choice. Some styles of programmed instruction, for example linear programming, might make the learning process dull and monotonous. Program learning, especially branch programming, can be expensive to develop. Most importantly, program development is an extremely time-consuming task. Also, this method of instruction is based on the view that knowledge is comprised of the aggregates or discrete elements. Consequently, the integration of an entire body of knowledge may sometimes become difficult. Some experts are of the opinion that the use of small sequential frames fails to create interest and challenge talented students in the group. Now you can try to answer the following questions. 1. What is the difference between mass instruction and individualized instruction? 2. What do the term program and frame refer to in program learning? 3. What are the things to consider when selecting a unit? 4. Which are the 3 steps in the construction stage of program development? 5. What are the major limitations of program learning? Now you may go through the reference books given here. Principles of Education by Chandra S.S. and Sharma R.K. in 1996, Atlantic Publishers, New Delhi. Sociology of Education by Chandra S.S. and Sharma R.K. in 1996, Atlantic Publishers, New Delhi. Methods and Techniques of Teaching by Kochar S.K. 1977, Sterling Publishers, New Delhi. Teaching of Social Studies by Mangal S.K. and Mangal U. in 2008, PHI Learning, New Delhi. Teacher Education by Mohan R. 2011, PHI Learning, New Delhi. Changing Attitudes to Education in India by Reddy K.V. 2002, Atlantic Publishers, New Delhi. Advanced Education Technology by Sharma R.N. and Chandra S.S. 2003, Atlantic Publishers, New Delhi. Thank you for watching this program. We can meet again with another topic. Have a nice day.
Agni, the fire, is the most potent source of light and heat energy. Since hundreds of millennium, fire has significantly contributed to the progress of humankind on the path towards civilization. It has banished darkness and provided knowledge, helped shape things of daily use, and aided societies to keep on the move. How did fire come into being? And how did man learn to use it? There are no definite answers to these questions. Blazes ignited by volcanic conflagration and lightning are speculated to have served as man's first source of fire. From such terrifying blazes, primitive man is believed to have kindled his own fire over 5 lakh years ago and gradually become conversant in using it for various purposes, such as to keep himself warm, to hunt animals, and to cook food. Much later, in the Neolithic age, he used fire to clear forests for agriculture and human settlements, and also learned to make fire by striking flints together, or by using simple tools such as the fire saw, the fire plow, and the fire drill. Since then, the application of fire has brought about significant changes in man's ways of living. In particular, it was the application of fire in pottery, in extraction of metals, and in their conversion into alloys that led to the emergence of the Metal Ages. For the growth of civilizations in ancient Greece, Egypt, and the Indus Valley, and of course for the industrial revolutions of the last few centuries, fire has immensely contributed. But fire, which has benevolent and destructive powers, was a mystery to the ancient people. They believed it to be of heavenly origin. According to Greek mythology, fire was stolen by Prometheus for the good of his fellow beings. The Persians interpreted fire as a true gift of the gods, fit to be venerated and worshipped.
Agnimile purohitai yajñasya deva mritwijam hotaram ratnadhatam aggaswaha.
The Vedic societies looked upon Agni the fire as a divine being who resides everywhere, despising none. He is the possessor of power and wealth and the divine messenger between the god and human beings. As did the Vedic mystics, the Greek philosophers, Plato and Aristotle, thought fire to be an element essential for the constitution of life and matter. Such mystical ideas prevailed until the later 18th century, by which time scientists had discovered the essentiality of air for the occurrence and persistence of fire. Antoine Lavoisier, in 1772, showed that an active fraction of air combines with burning substances, making them weigh more than they initially did. Fire, he explained, is a result of a chemical reaction between oxygen and flammable substances.
The complex phenomenon of fire is basically a manifestation of combustion. Combustion can be defined as an extract of oxygen from the air, producing combustion products along with the emission of heat and light in the form of flame.
Not all combustions, however, result in fire. Often oxygen unites with some substances at such a slow rate that little heat or no light are given off. Such a slow reaction is called a slow combustion rather than burning, which is very rapid. A good example is the rusting of iron.
Before we go into the details of chemical reactions involving flammable vapors and oxygen that yield fire, let us look at the conditions needed for fire to occur.
First, there should be a fuel. Most fuels are complex hydrocarbon compounds. A fuel could be a solid such as coal or wood, a liquid such as oils, and a gas such as methane. Second, there must be plenty of oxygen. Third, the fuels must be heated to ignition temperature at which they undergo thermal decomposition, releasing flammable vapors with which oxygen unites rapidly in a chemical reaction. In fact, all fuels, except gases, must first be converted to their gaseous state for them to yield fire. It is therefore that kerosene, a liquid fuel, though exposed to air, will not catch fire even when a pilot flame is brought in contact with it. However, upon heating directly to its ignition temperature, kerosene converts into its vaporous state. Now, when a burning stick is brought close to the vapors, they combust readily into a flame. In the case of combustible solids, heat decomposes involatile fuel molecules into tiny volatile molecular fragments which react with oxygen of the air to yield fire, producing a series of combustion products.
Let us investigate chemical reactions by taking the example of methane and oxygen.
Each molecule of oxygen contains two oxygen atoms which are bonded together. Methane is a natural gas. Each of its molecule contains one carbon and four hydrogen atoms bonded together in a particular arrangement by a force arising from electron sharing, which accounts for the molecular energy. When methane is ignited, the bonds break, freeing carbon and hydrogen atoms, which then react separately with oxygen. One carbon and one oxygen atom unite to form carbon monoxide. One oxygen and one hydrogen atom combine to form the hydroxyl radical. While carbon monoxide combines with one oxygen atom to form carbon dioxide, the hydroxyl radical unites with one hydrogen atom to form water. These products of combustion then escape into the atmosphere.
The total energy of combustion products tends to be less than those of the fuel and oxygen involved in combustion. The difference in energy gets released as the heat of the fire. It is therefore that combustion reactions producing fire are exothermic chemical reactions.
Let us now take the case of wood, a solid fuel, the bulk of which is cellulose and water. Lignin, resins, and inorganic substances form the rest of the woody matter.
Wood, which produces upon burning carbon dioxide and water, contains cellulose, which is a polymer of glucose units. Each unit contains 6 carbon, 10 hydrogen, and 5 oxygen atoms. Wood upon burning also yields charcoal and ash, which are a mixture of unburnt carbon particles and inorganic substances. In the fire, where do these extremely complex chemical reactions occur, producing a series of combustion products?
The complex reactions between fuel and oxygen occur in the flame zone. A flame is recognized as a glowing body of oxygen and fuel in their vaporous or gaseous state, undergoing the process of combustion, producing heat and light.
In some burning systems, the combustion of fuels tend to be incomplete due to insufficient supply of oxygen. This results in a luminous yellow flame as that of a matchstick.
Some fuels can undergo complete combustion when there is sufficient supply of oxygen for the fuel to burn completely. Such burning systems produce a blue flame. The flame of the Bunsen burner is a good example.
Based on this difference, flames are usually classified as either diffusion flames or premixed flames. Diffusion flames are those flames in which oxygen needed for combustion diffuses from the surrounding air and therefore meets the fuel only at the flame.
Let us learn more about diffusion flames by examining the flame of a candle.
When a candle is lit, the heat of the matchstick melts the wax, which is a hydrocarbon compound. The molten wax is carried up the wick and vaporized. The vapors finally burn in a diffusion flame containing 4 distinct zones. Around the wick is the dark non-luminous region containing volatile unburnt wax vapors. Hugging the dark zone is a yellow brightly luminous zone where decomposition of hydrocarbon molecules takes place. Some of the hydrogen and carbon monoxide produced by decomposition react with available oxygen to form water and carbon dioxide respectively in this zone. The free carbon particles produced in this zone are responsible for the diffusion flame's yellow luminance and sooty nature. Soot is nothing but unburnt carbon particles. Outside the luminous zone is the third faintly visible non-luminous zone, where some of the carbon monoxide, carbon and hydrogen undergo a final phase of reaction with oxygen of the air. Lastly, there is a fourth blue zone at the base of the flame. Here some of the carbon monoxide formed in the luminous zone undergoes complete combustion by reacting with plentiful oxygen to form carbon dioxide.
Let us now examine the case of premixed flames, which result from those burning systems where fuels are mixed with oxygen prior to their entry into the flame zone.
The flame of the Bunsen burner is a good example of a premixed flame. Named after Robert Wilhelm Bunsen, a German scientist, the burner consists of a metal barrel into which gaseous fuel is injected under pressure through a nozzle. To provide appropriate quantity of oxygen needed to burn the gaseous fuel completely, the barrel is equipped with air holes, through which air gets sucked into the barrel where it mixes with the gaseous fuel. On ignition, this fuel-oxygen mixture burns with a hot, soot-free, predominantly blue flame having 3 distinct zones. The inner zone, close to the mouth of the burner, is dark because it has cold, unburnt mixture of gaseous fuel and oxygen. Therefore, a stick placed in this zone readily burns on the 2 sides touching the blue flame, while the central portion in the dark zone remains unburnt. Bright bluish-green mantle, a zone of chemical reaction. Here, the hydrocarbon molecules break into free hydrogen and carbon. These react quickly and rapidly with a premixed, abundantly available oxygen to form carbon dioxide and unstable carbon monoxide and hydroxyl radicals. The unstable products undergo further reaction in the outermost pale bluish and turbulent flame zone, yielding carbon dioxide and water vapor. As all the fuel gets consumed without any loss, these carbon-free, non-sooty and non-luminous flames produce a high temperature Therefore, premixed flame systems have several industrial and domestic applications. One such example is a domestic gas stove into which a fuel, that is, liquefied petroleum gas, is fed through a pipe underneath the stove. On the pipes are air ports through which air enters and oxygen present in it mixes with the fuel, which emerges from the tiny holes of the burner. On ignition, the mixture burns in a soot-free blue flame with a temperature of over 1000 degrees centigrade. Still hotter flames, required for special purposes in industries, are obtained by burning gaseous fuels such as acetylene mixed with oxygen under pressure. The oxyacetylene mixture, when burnt, gives out a premixed flame with a high temperature of over 2300 degrees centigrade. The mystery that is Agni, the fire, is no longer an unsolved phenomenon. Quite simply, it is a physical manifestation of combustion that helps man harness the bounties of the earth as well as those of the heavens. With thousands of everyday uses, turning the proverbial darkness of human existence into a celebration of light.
Welcome to the session of Organization of Knowledge in Libraries, Library Classification. In this session, we will discuss the remaining part of normative principles, canons for work in the idea plane. Canons for chain. The process of division of knowledge should proceed from general to specific, forming a sequence of subordinate classes. This sequence of subordinate classes is called a chain of classes. It leads us to an ultimate class from an original universe. The formation of chain of classes should be properly regulated. Each chain of classes must satisfy the requirements of the two canons. First one, canon of decreasing extension. The canon states, while moving down a chain from its first link to its last, the extension of the classes or the ranked isolates, as the case may be, should decrease and the intention should increase at each step. The term extension denotes the scope of a subject. It is measured by the number of entities comprised in the class or by the area covered by the subject. Intension refers to the specificity of the meaning of a class. It can be determined by the number of characteristics used to derive the class from the original universe. As an example, zoology is a class of greater extension, whereas cat is a class of smaller extension, but greater intention as shown on the screen. Zoology, vertebrates, mammals, cat family, domestic cats. Domestic cats is a subdivision of zoology arrived at by applying 4 characteristics successively. We can see that the above chain satisfies the canon of decreasing extension. Second one, canon of modulation. This canon states a chain of classes or of ranked isolates should comprise one class or one ranked isolate, as the case may be, of each and every order that lies between the orders of the first link and the last link of the chain. Canon of decreasing extension explains how do we form a chain. Canon of modulation insists what should be included in a chain. Application of the canon can be made clear by the following illustration. Suppose we derive a chain of geographical entities considering world as the first link, original universe, and Thenipalam as the last link. In the progressive classification of the geographical area world, we arrive at Thenipalam by using all the required characteristics. The chain shown on the screen contains all the links between the first and the last. Entity, world, link 1, characteristic used, original universe. Entity, Asia, link 2, characteristic used, by continent. Entity, India, link 3, characteristic used, by country. And finally, entity Thenipalam, link 7, characteristic used, by village. This chain is a modulated chain. Even if we exclude one of the links between the first and the last, the remaining entities result into a chain and it satisfies the canon of decreasing extension but violates the canon of modulation. Violation of the canon of modulation will result in missing links in the progressive subordination. This will create problems in subject cataloging and indexing. Canons for filiatory sequence. In the progressive classification, each class will be coordinated to some other classes and at the same time it may be a subordinate or superordinate link of some other classes. This situation is called the array and chain relationships among classes. The two canons for filiatory sequence insist that a scheme of library classification should clearly identify both coordinate and subordinate classes of any class and they should be arranged among themselves according to their filiatory relationship. The first canon corresponds to subordinate classes and the second is related to coordinate classes. First one, canon for subordinate classes. According to this canon, when the array and chain relationships among classes are transferred into a linear sequence, the subordinate classes should immediately follow their superordinate class. Second, canon of coordinate classes. This canon says that in a coalesced array, two consecutive classes should not be separated from each other by any class other than the subordinate classes of the first class. Let us assume that the original universe is denoted by 0 and the classes of order 1 are A, B, C. Let us also assume that the classes of order 2 are A1, A2, A3, B1, B2, C1, C2, C3. Further, there are classes of order 3 which are denoted as B11 and B12. Then the coalesced array would consist of the various classes in the following sequence: 0, A, A1, A2, A3, B, B1, B11, B12, B2, C, C1, C2, C3. According to the canon of subordinate classes, B1, B11 and B12 should immediately follow class B as shown on the screen. According to the canon of coordinate classes, B1 and B2 should not be separated from each other by any class other than B11 and B12, having B1 as their common immediate universe. principles of helpful sequence in an array. The canon of helpful sequence insists that some guiding principles should be used to arrange the isolates in an array. Dr. Ranganathan enumerated the following principles for this purpose. First one, principle of later in time. If the subjects in an array of subjects or the isolates in an array of isolates have originated in different times, they should be arranged in a parallel progressive time sequence except when any other overwhelming consideration rules it out. Second, principle of later in evolution. If the subjects in an array of subjects or the isolates in an array of isolates belong to different stages of evolution, they should be arranged parallel to the evolutionary sequence except when any other overwhelming consideration rules it out. Third, principle of spatial contiguity. If the subjects in an array of subjects or the isolates in an array of isolates occur contiguously in space, roughly along an unidirectional linear or a radial line or a circle, they should be arranged in a parallel spatial sequence except when any other overwhelming consideration rules it out. This principle includes really a collection principles such as 1. Principle of bottom upwards or top downwards. 2. Principle of left to right or right to left. 3. Principle of front to back or back to front. 4. Principle of clockwise or anti-clockwise direction. 5. Principle of periphery to center, center to periphery. 6. Principle of away from position. It can be seen that the first 5 occur in antithetic pairs. First one, principle of bottom upwards or top downwards. If the subjects in an array of subjects or the isolates in an array of isolates can be conveniently taken to occur along a vertical line, they may be arranged from bottom upwards or from top downwards according to the helpfulness. Second, principle of left to right or right to left. If the subjects in an array of subjects or the isolates in an array of isolates can be conveniently taken to occur along a horizontal line, they may be arranged from left to right or right to left according to the helpfulness. Third, principle of front to back or back to front. If the subjects in an array of subjects or the isolates in an array of isolates can be conveniently taken to occur along a horizontal line, they may be arranged from front to back or from back to front according to the helpfulness. Fourth, principle of clockwise or anti-clockwise direction. If the subjects in an array of subjects or the isolates in an array of isolates can be conveniently taken to occur along a circular line, they may be arranged in the clockwise direction or in the counterclockwise direction according to the helpfulness. Fifth one, principle of periphery to center or center to periphery. If the subjects in an array of subjects or the isolates in an array of isolates can be conveniently taken to occur along a radial line of a circle or cylinder, they may be arranged from periphery to center or from center to periphery according to the helpfulness. Sixth one, principle of away from position. If the subjects in an array of subjects or the isolates in an array of isolates can be conveniently taken to start from a certain point and diverge away from it roughly along a line, they may be arranged from the starting point along a diverging line if it is helpful. Geographical contiguity. The division of the surface of Earth, such as continents, countries, states, districts, etc., lie on a surface and not on a line. One division may lie adjacent to several other similar divisions. Therefore, it is not easy to arrange the geographical areas according to the principle of spatial contiguity. More than one principle may have to be used in each context. Fourth one, principle of quantitative measure. If the subjects in an array of subjects or the isolates in an array of isolates admit of quantitative distinction, they may be arranged on the basis of their quantitative measure, that is, on the basis of increasing quantity or decreasing quantity according to the helpfulness. Fifth, principle of increasing complexity. If the subjects in an array of subjects or the isolates in an array of isolates show different degrees of complexity, they should be arranged parallel to the sequence of increasing complexity, except when any other overwhelming consideration rules it out. Sixth, principle of canonical sequence. If the subjects in an array of subjects or the isolates in an array of isolates are traditionally referred to in a specific sequence, although no underlying principle is discoverable, it will be convenient to conform to this traditional sequence. Sometimes no specific characteristic can be observed as forming the basis for the division of classes, but the classes are simply those into which their immediate universe had been traditionally divided. For example, the first order division of few main classes like mathematics, physics, geology are arranged according to this principle in the Collin classification. These are called the canonical classes. Seventh, principle of literary warrant. This principle suggests that the subjects in an array of subjects or the isolates in an array of isolates may be arranged in the sequence of the decreasing quantity of the documents published or anticipated to be published on them, except when any other overwhelming consideration rules it out. The application of this principle requires care and judgment. In an international classification scheme, it should be applied without any bias to the country of origin of the scheme. Eighth one, principle of alphabetical sequence. When no other sequence of the subjects in an array of subjects or the isolates in an array of isolates is more helpful, they may be arranged alphabetically by their names current in international usage. Generally, alphabetical sequence is not helpful. Hence, the application of it should be restricted to a context in which no other sequence is more helpful and relevant. In such a situation, the simplicity of alphabetical sequence can be exploited. This principle is normally used in many temporary and limited contexts. For example, the students in a classroom or the participants in a meeting can be arranged using this principle. However, in a permanent and universal context, such as in the classification of subjects, its use has to be restricted. The preconditions for the use of the principles are: 1. No other better sequence is available. 2. The universe to be classified should have an internationally accepted nomenclature. Sometimes few other principles are also employed for arranging the isolates in an array. They are listed below as additional principles of helpful sequence. Ninth, additional principle of helpful sequence. First point, concreteness, abstractness sequence. Second, extension intention sequence. Third, developmental sequence. Fourth, naturalness artificiality sequence. Fifth, dependency sequence. Wall picture principle, act and action, actor tool principle, cow-calf principle. Now let us summarize the whole session. In this session, we studied the canons for chain and the canons for filiatory sequence. We also discussed the guiding principles that can be used to arrange the isolates in an array to achieve a helpful sequence among them. Here are a few assignments for you to work out. First one, State and explain the canon of modulation. Second, write a short note on canon of decreasing extension. Third, describe the principles of helpful sequence with suitable examples. Fourth, critically examine how far Collin classification observes the principles of helpful sequence in arranging the isolates in the main classes. Now I will provide you with few books for further reading. Theory of Classification, 4th edition, revised, written by Krishnan Kumar, published by Vikas Publishing House, New Delhi, 1988. Library Classification, Facets and Analysis, written by Hussain Shabahat, published by Tata McGraw-Hill, New Delhi, 1993. Third, Prolegomena to Library Classification, 3rd edition, written by Dr. Ranganathan S.R., published by Sharda Ranganathan Endowment for Library Science, Bangalore, 1967. Bachelor of Library Science Study Material, University of Calicut, 1995. Thank you for watching this session. See you in the next session. Until then, bye.
Hello and welcome to the Interface program. Today we have with us Professor R. Raj Rao, author, Professor of English Literature at Pune University and one of India's leading gay rights activists. Sir, welcome.
Thank you.
We will begin our discussion with your own writings.
I have been writing for almost 25 years now, so it's been a varied output. I have written poetry, short stories, novels, a biography of the poet Nissim Ezekiel, lots of critical writing and non-fiction, academic papers and newspaper columns, so it's been different things that I've been doing, different kinds of writing that I have done. My creative writing, of course, is poetry and fiction and also a few plays. This is what I regard as important, but the other work is equally important. The critical work is important. My biography of the poet Nissim Ezekiel still remains the only biography of a post-independence Indian poet in English. And then I've done some translations as well. A recent book of mine, a recent publication of mine is a translation of Lakshmi. Lakshmi is a transgender activist, and her autobiography, which first came out in Marathi, has been co-translated by me along with P.G. Joshi, and we've translated this into English, and Oxford University Press has just brought it out. So the different things I've done. And I think that that's how it should be. A literator must be able to practice different forms of writing and not just concentrate on one form. We're talking about multimedia, but within writing, I think there has to be a multimedia nature to it.
So the entry in Wikipedia about you begins almost like this. You are one of the leading gay rights activists in India. So, could you elaborate on the kind of activism that you are involved in?
See, there are different kinds of activism, and my activism is literary and academic It's not the activism of, say, people like Ashok Rao Kavi, who founded the Humsafar Trust in Bombay way back in 1990, or Bindu Madhav Khire, who founded the Sampathik Trust in Pune about 10-15 years ago, because that is grassroots activism. It has to do with field work and outreach work and so on. And but I'm not in that sector. I don't have a support group or anything. But I think that I think I've said this several times, that writing and activism are two sides of the same coin, not just for people like me, but for anyone who's concerned with with identity issues. Take Dalit literature. Take a writer like the late Namdev Dhasal. I'm sure it was the same for him, that his writing and his activism were two sides of the same coin. I guess it's the same for many women writers. So, activism, yes, but activism through art and literature. And this is something that people don't understand. Marginal literatures, I think, have often been misread as literatures that have an agenda, that literatures that are propaganda, and that literatures that are inferior in terms of artistic ability, and all this is not true. It has to be dispelled, because one can have issues about which one is writing, and one can also be artistic and literary. Even Commonwealth literature began with that, wouldn't you say a writer like B.S. Naipaul or Salman Rushdie, don't they have issues that they are tackling in their work? And yet they are part of the canon. Whereas with gay writing particularly, and I'm not the only one to be saying this, there are others who would endorse what I'm saying. Somehow the feeling is that if you call yourself a gay writer, then it means your politics are being foregrounded and your art is taking a back seat, and that's not at all true. So actually I'm not sure whether I like the word activist, because the moment you invoke a word like activist, this is what people think, that writing is only an excuse for making political statements. But that's not how it is. My training is that of a literary scholar. I teach in an English department. I've been teaching for more than 25 years. And I think that for me, literary values in a work are as important, if not more important, than what you're writing about. So that's my position. And also that if you take gay fiction, at the end of the day, it's about love. We don't make such a big issue when writers write about heterosexual love, which everybody is doing. So I don't even like to use the word gay sometimes, because it's love. In some cases it's heterosexual love, in other cases it's homosexual love, but it's love. It's a love story. My novel, The Boyfriend, is a love story. My second novel, Hostel Room 131, is also a love story. My forthcoming novel, Lady Lolita's Lover, is also a love story. If the characters concerned belong to the same gender, that's just an accident. That's how I see it.
So, when we talk about your boyfriend, it deals with homoeroticism in a very apparent way, and it almost borders onto pornography or something like that. So, how will you respond to such a?
No, I'm very happy if you say that, because I don't have a moral position vis-a-vis pornography. I don't, if I say pornography is bad, then I'm assuming a very moral positionality, which is not what I do. I have frequently said that gay writing needs its own aesthetic. I recently said this at a conference, that gay writing does need its own aesthetic, and what that aesthetic would entail is also sexual explicitness, right? Now one can go into the reasons for that and ask why gay love stories are more sexually explicit than heterosexual love stories, though I'm not sure that that's always the case. But it is certainly part of my aesthetics, and so if you say verges on pornography, that makes me happy. It doesn't offend me. I don't believe in censorship and self-censorship, and I think that one must write as freely as one can, without bothering about offending people and so on, without worrying too much about worrying if something is obscene or vulgar or so on. I think that one has to write, and sex is very much a part of life. So to say that you can't express it in a novel or in a short story or in a poem, I think is being a hypocrite. So I'm totally against self-censorship, where writers think that, well, this is offending. And of course we are living in times where you've got to be careful, because I think we are living in terrible times as far as censorship is concerned, and you don't want to do things that might get you into trouble for no reason, if it can be avoided or if it can be said in a roundabout way. That's a nice thing about English, incidentally, that a lot of the time you get away saying what you want to say, because the people who are likely to take offense don't even understand what you're saying. With the regional languages, they don't have that luxury, because whatever they say is immediately taken at face value. But no, I mean, pornography is all right. I'm not making a distinction between pornography and non-pornography. If I want to write a sex scene, I'll write a sex scene. And if my publishers and editors don't have a problem with it, and they let it be, then I think that it's nobody else's business to worry about it.
Okay, so you consider it as a matter of openness of the society?
Yes, yes. The other thing I wanted to say is that when I write a homosexual novel or short story or play or poem, I'm not self-conscious about it, and I'm not apologetic about it. There's a lot of gay writing, particularly in the regional languages, which tends to be apologetic. So it operates at a level where, the person concerned is trying to come out to parents, is trying to come out to friends, and is having a lot of trouble doing that. My work is not like that. My characters are sort of in your face gay, and they realize that just as some people are straight, other people will be gay. So there's no point in being apologetic and and being self-conscious about it. So that's partly why you might read it as pornography, because it is in your face. If you take The Boyfriend, UD is cruising. The very first chapter is called Gentleman, and it's set in a train station washroom in Bombay, the Churchgate station. And, that has been a hot cruising spot for a long time, and he's cruising there, and, he's not worried. Actually, he lives with his mother, but his mother has gone someplace, and he picks up a boy and takes him home because there's no one at home. But he's not self-conscious, or he's not guilt-ridden. What if my mother comes to know? And so I think that for me, being gay also has to do with decentering the idea of normativity. This whole business of being honest and being good and being monogamous and being faithful and being dutiful is a normative idea. So my stories unfold in a way which proves that for me the important thing is to dismantle the idea of normativity.
Professor Rao, you are one of the leading champions of LGBT writing. So could you tell us something about the present position of LGBT writing and its place in the Indian syllabus? And maybe you must have worked, you must have put in some work to bring that into Indian syllabus, no?
Yes, there's a fair amount of LGBT writing by this time. Incidentally, LGBT stands for lesbian, gay, bisexual and transgender. And by this point, there's a fair amount of it. There are two major anthologies which were brought out by a leading publisher, Penguin, in 1999. So that's a while ago. Yarana and Facing the Mirror. Yarana is gay writing from India and Facing the Mirror is lesbian writing from India. But about 3 years ago, another anthology of queer writing came out. It was called Out, Stories from the New Queer India. And there have been full-fledged novels, there have been my novels, but there have been other novels and short stories as well that people have been writing. So I think the body of gay writing and lesbian writing will, it has existed and it will continue to exist. Then there has been the other kind of critical writing, of which there's also been quite a lot until this time. And, these are by mainstream scholars. There's the book Phobic and Erotic. There's the book Same-Sex Love in India, readings from literature and history, edited by Ruth Vanita and Salim Kidwai. There's the book Because I Have a Voice. There's the book Forbidden Sex Texts, written by Hoshang Merchant. And all these were published by major publishers. The publishers are Macmillan and Penguin, Routledge, Seagull. So the two kinds of writing go on simultaneously. There is the literary representation of same-sex love in the fiction and the poetry that we have, and then there's also the theorizing of it in these other books that I mentioned. So what happens is that by this point, there's also my own book, Whistling in the Dark, 21 Queer Interviews, which is made up of interviews with LGBT people. And the nice thing about that book is that it talks to people across a spectrum of values. So there are university professors, but there are also people who work as, say, auto drivers and so on. That book was almost done the way a sting operation is carried out, and my publisher, Sage, at that time was very cooperative. They allowed the book to go to press without even getting sort of written consent from some of the respondents, because we changed their names and so on. And we pointed out to them, to the publishers, that since we were asking very personal questions, people wouldn't give their consent. So we sometimes didn't even tell them that we were interviewing them for a book and so on. So there's all that work. So what happens at this point is that there's enough fiction, poetry, drama. There's Mahesh Dattani's plays as well, which I forgot to mention. And there's enough theoretical material available to start a very good university course at the PG level or even at the UG level. And in my own university, the University of Pune, I have been offering the course that I call it Queer Theory and Literature, but the syllabus calls it Alternative Literature. 2. Alternative Literature. 1 is Dalit writing, Dalit Sahitya. The University of Hyderabad has been offering a course for a long time. I think Kalyani in West Bengal, because wherever there are LGBT people on the faculty, you can be sure that they will want to start a course like that. So there's Niladri Chatterjee at Kalyani, there's Hoshang Merchant in Hyderabad, there's me in Pune, and then other universities. JNU, I think, has had several dissertations on LGBT studies. Delhi University, the major universities and even the smaller ones, I think, are catching on. The thing about research is that, one is obliged to look for new things to research. And in a way, it's a bit opportunistic because people just want to latch on to the bandwagon. So there's a lot of research going on which is not coming from conviction or anything. It's just that researchers are desperate to look for new areas of research, But the end result is that there are courses that are being taught, there's research at the MPhil and PhD levels, and that's going on. All this is a good thing. And I think the future for LGBT studies in Indian universities is bright. In Western universities, it has already been around for a long time. But Indian universities are finally, I think, catching on, and I think the years to come will see more and more work in this direction.
Professor Rao, there's something that's called cultural wars that has taken place in American universities, no? So is there something that is taking place, something that's similar to that in Indian universities with regard to the maybe inclusion of Dalit literature, LGBT studies and all that? Do you find some kind of resistance, or you find the opening up a very easy process?
No, it may not be easy. Like I just said, if it's visible now to a greater extent than it was visible earlier, it's partly because of the opportunism of teachers and students who are looking for new areas to research. This does not necessarily mean that they've overcome their own conservatism. And that's what I meant by saying that there's a bit of hypocrisy here, because they may be conservative and yet they find that they have to be open to new areas of learning and new systems of knowledge and new kinds of research. Also, furthermore, the change happens more in the cities than in the smaller universities. So the universities that I mentioned are, of course, the metro universities. In the smaller universities, in the undergrad colleges, I'm not sure whether the same changes are reflected there. Because academia tends to be conservative. Academia, I think, is the slowest to take note of changes that go on around us. I mean, there are English departments that are still stuck with Shakespeare and, the writers of the English literary canon, and they are completely unwilling to go beyond that and explore new areas and new territories. So it's varied. I don't think in a country as vast as India you can generalize. There would be universities which are very forward-looking and even radical, but equally there would be lots of other university departments of English which are extremely conservative. This conservatism is a result of lethargy on the part of teachers who are unwilling to acquaint themselves with newer things. I mean, I'm sure there are lots of teachers who still depend on their own notes which they took during their own student days, and this is what they do in the class. So there's all kinds of universities, and I don't think one can generalize, but a change has been made, a beginning, not a change, a beginning has been made, and I hope, I'm optimistic that as the years go by, we'll see a greater willingness. Let me put it that way. We'll see a greater willingness on the part of teachers and students to acquaint themselves with new things.
Professor Rao, from the academic side, we will move on to the social side. Recently, the Supreme Court of India has again recriminalized same-sex love and same-sex intercourse and things like that. So how will you comment on such a verdict and its impact on the Indian society?
I think so much has been written about this in the newspapers, in the media. I myself write a weekly column for a tabloid newspaper in Pune, and frequently talked about this judgment and its implications. needless to say, it's extremely unfortunate that a judgment like that came out. I might tell you that nobody expected it. Everybody thought that the Supreme Court would uphold the Delhi High Court judgment of July 2009. So I think it came as a big blow and a big shock to all concerned, including people in the legal profession. I was reading a book called Courting Injustice. It's by a lawyer by the name of Rajesh Talwar, and he was one of the first people to file a petition in the Delhi High Court for the scrapping of Section 377, even before the Nas Foundation did it. And his book came out before the 2013 judgment. And he says somewhere in the book that, he almost takes it for granted that the Supreme Court will uphold the verdict of the Delhi High Court. So when that 11th December judgment of 2013 came out, it was a big shock to all of us and a very unfortunate thing to happen. So what can one say? I mean, Supreme Court judgments are final and binding. They can't be challenged, although there is a curative petition pending in the Supreme Court to rehear the case. And that in a way is the only option now. The government certainly isn't going to do anything about changing the law. So that's where we are, and it's a big setback. I mean, there's nothing more I can say, because you can't appeal against a Supreme Court judgment, and it's a big setback because technically it even makes an interview like this a criminal activity. Because if a certain kind of love is illegal, then talking about it and writing about it should also be illegal. So that's where we are. I mean, I think it's a stalemate, and we are up against a wall, and I don't know.
Okay, Professor, thank you so much.
Thank you. I enjoyed giving you this interview, and I hope it's useful to the students who will see the program. Thank you.
Hello and welcome to the session on important biological databases. Databases are convenient systems to properly store and manage any type of data from various diverse resources. They can be considered as electronic filing cabinets to store vast amount of information. Today, biology has developed into a data-rich science, and as the volume of genomic data increases, the need for computational methodologies to store and communicate these large data sets has also grown tremendously. Thus, the major challenge in the genomics era is to store and handle this huge volume of information through the organization and use of computer databases. The development of databases is to handle the vast amount of molecular biological data is thus a fundamental task of bioinformatics. Thus, the biological databases have come into existence as invaluable tool to serve scientific community. A biological database can be defined as a large organized body of persistent data, usually associated with computerized software designed to update, query, and retrieve components of the data stored within the system that make biological data available to scientists in computer readable form. They provide automated storage and retrieval of experimental data and contain information from research areas including genomics, proteomics, metabolomics, microarray gene expression, and phylogenetics. They also help to understand the biological phenomena from the structure of biomolecules and their interaction and the whole metabolism of organisms, and to understand the evolution of species. There are different types of biological data, and so there are different types of databases. They depend on the nature of information being stored, that is, whether the sequence information or structure, images of 2D gel, macromolecule structures, organism specific information, metabolic pathways, and also information from literature. These databases are either with proprietary rights or public domain databases with free access. This information is stored either in flat file or it can be relational database. Using the web interface of the databases, user can retrieve the information of their interest, providing the appropriate keywords. Most of the biological databases provide the facility to save the search result locally on a computer. Cross-referring between these databases helps to move from one database to another. The chief objective of the development of a database is to organize data in a set of structured records to enable easy retrieval of information. Although data retrieval is the main purpose of all databases, biological databases also identify connections between pieces of information that were not known when the information was first entered. For example, databases containing raw sequence information can be used to identify sequence homology or conserved motifs. These features facilitate the discovery of new biological insights from raw data. The biological databases works with the help of a computer program, that is Database Management System or DBMS. These are a collection of programs that enable to store, modify and extract information from a database. It helps to manage and query the database to provide answers to the questions. A database can be queried in different possible ways, such as giving a sequence or fragment of a sequence and thus finding sequences in the database that are similar to it, giving a sequence of a protein of unknown structure to find structure in the databases that are similar to it, give a protein structure or fragment to find protein structures in the databases that are similar to it, give a sequence of a protein structure to find sequence in the data bank that corresponds to similar structures. Types of databases. There are different types of databases based on the information they store. Biological databases can be broadly classified into three groups: sequence database, structural database, and specialized databases. Sequence database. Nucleotide sequence database, for example NCBI, EMBL, DDBJ. Protein sequence database, for example SwissProt, PIR. Structure database, for example PDB, MMDB. Specialized databases, for example MGI, RGP. Sequence database. A sequence database is a repository of all available nucleotide sequences or protein sequences which are determined by DNA sequencing and protein sequencing. These databases help us to retrieve related sequences by homology searching. The sequence database also forms a backbone resource from which other value-added databases are developed. Because there are multiple databases for the same type of data, and because there are identical sequences in the same database, efforts are now being made to construct a non-redundant set of sequences from multiple sources. The sequence database consists of both primary and secondary databases. Primary databases or raw database contain original submission of protein and nucleotide sequences from experiments and are controlled by the submitter. The primary nucleotide sequence databases are NCBI, EMBL and DDBJ, and the primary protein sequence databases are SwissProt and PIR. Secondary databases or derived databases contain data that are derived from the annotation, curation and analysis of data in the primary databases. These are usually subsets of the primary databases. Information like secondary structure, ESTS, hydrophobicity plots and domains are stored in the secondary databases. Major secondary databases are ProCite, PRINTS, PFAM-TEG. Primary nucleotide sequence database. Nucleotide sequence databases organize and distribute nucleotide sequences from all available sources. They contain sequences submitted directly by scientists and genome sequencing group and sequences taken from literature and patents. The International Nucleotide Sequence Database consists of the three primary nucleotide sequence databases. 1. GenBank, that is National Center for Biotechnology Information. Number 2. EMBL Nucleotide Sequence Database or European Molecular Biology Laboratory. Number 3. DDBJ or DNA Data Bank of Japan. GenBank is physically located in the USA and is maintained by National Center for Biotechnology Information and accessible through NCBI portal over Internet. EMBL or European Molecular Biology Laboratory is in UK and DDJB or DNA Data Bank of Japan is in Japan. The three primary databases are repositories for nucleotide sequence data from all organisms. They accept nucleotide sequence submissions and then exchange new and updated data on a daily basis to achieve optimal synchronization between them. Historically, the three started out as separate databases with independent means of collecting the data and with different formats for arranging it. Now, the three together form the International Nucleotide Sequence Database collaboration. They have uniform format and exchange data on a daily basis with the result that it is sufficient to connect to any one of them in order to access the sequences present in all the three. The multidisciplinary research group consists of scientists from diverse fields who work on the management of biological data as well as to develop bioinformatic tools to analyze biological data. Data can be exchanged between the three collaborating organizations on a daily basis. The nucleotide databases have reached such large sizes that they are available in subdivisions that allow searches or download that are more limited and hence less time consuming. For example, GenBank has currently 17 divisions. There are no legal restrictions on the use of the data in these databases. However, there are patented sequences in the databases. Having a sequence in the database means it is readily available to the scientific user community. A repository of primary nucleotide sequence data that is freely accessible is essential for computational analysis and genome research. DNA sequence information can be obtained at the following three levels. Genomic sequence level, which include information at the level of untranslated sequence, exons, introns, mRNA and cDNA, that is transcription level, protein, that is translational level. Thus, DNA databases contain a variety of different types of data. The genomic sequence data can be further divided into three categories: full-length CDs, partial sequences including EST and STS, genomic sequences including HTG and GSS, all of the rest sequences which include SNPs. The ESTs and other partial sequence data such as sequence tagged sites or STS are usually short sequences, whereas full-length sequences making up the rest of the database tend to be much longer. The description of precise location, size and function of sequence elements within the DNA, RNA or protein and the prediction of function from sequence refers to annotation. A genome must be annotated to provide the putative start, stop and structure of each gene along with predicted gene products and about similarities to other known or predicted genes and proteins. The Genomic Single Sequence Database or GSS contains single pass genomic sequences, exon trapped genomic sequences and ALUI PCR sequences. HTG or high throughput genomic sequence database contains unfinished DNA sequences. NCBI. The National Center for Biotechnology Information (NCBI) was established in November 4, 1988 as a part of the United States National Library of Medicine, a branch of the National Institutes of Health. The NCBI is located in Bethesda, Maryland. The NCBI houses genome sequencing data in GenBank and an index of biomedical research articles in PubMed Central and PubMed, as well as other information relevant to biotechnology. All these databases are available online through the Emmrc search engine. NCBI is directed by David Lippman, one of the original authors of the BLAST sequence alignment program and a widely respected figure in bioinformatics. What does NCBI do? NCBI creates public databases, conducts research program in computational biology, develops software tools for analyzing genome data, and disseminates biomedical information, all for the better understanding of molecular process affecting human health and disease. NCBI develops biomedical database, promotes standards for databases, data deposition and exchange. To help the scientific and medical communities, they develop, distribute, support and coordinate access to a variety of databases and software. They also conduct research on fundamental biomedical problems at the molecular level using mathematics, statistics and computational methods. In October 1992, the NCBI, the largest biomedical research facility in the world, took liability for the GenBank. In addition to GenBank, NCBI also supports and distributes a variety of databases for the medical and scientific communities. These include the Online Mendelian Inheritance in Man or OMIM, the Molecular Modeling Database MMDB of 3D protein structures, the Unique Human Gene Sequence Collection UniGene, a gene map of the human genome, the Taxonomy Browser and the Cancer Genome Anatomy Project or CGAP in collaboration with the National Cancer Institute. GenBank. GenBank was set up in 1979 at the LANL, Los Alamos. Since 1992, GenBank is maintained by the NCBI Bethesda, USA. It is the most comprehensive and annotated collection of DNA sequences and is a part of International Nucleotide Sequence Database Collaboration INSDC, which consists of DDBJ, EMBL and NCBI. Out of the three nucleotide sequence databases, GenBank is the major contributor of sequence data. A new release of GenBank is made every 2 months. Sequence information are incorporated from direct author submissions, large sequencing projects, the Genome Sequence Database, Santa Fe, USA, the United States Patent and Trademark Office, USTPO, and other international patent offices, and via exchange from partners of the INSD. Direct submissions to GenBank are prepared using one of two submission tools, Bankit or Sequin. Information can be retrieved from GenBank using NCBI's Emmrc retrieval system. GenBank may also be searched with query sequences using NCBI's web interface to the BLAST suite of programs. Each entry consists of a number of keywords, associated sub-keywords and an optional feature table. How to retrieve information from NCBI? The web link to NCBI is www.ncbi.nlm.nih.gov. From the drop-down, select Protein to retrieve protein sequence data. Then select Nucleotide to retrieve the nucleotide sequence data. Finally, type the protein name or the accession number and click Search. Next, select any one of the entry. If the query is the NCBI accession ID, the details of that particular ID will be displayed directly as shown here. Each entry consists of a number of keywords, associated sub-keywords and an optional feature table. The keywords include definition, a concise description of the sequence, for example lipase from Pseudomonas fluorescens. Accession, accession number is a unique constant code assigned to each entry, for example M13667. Version, this indicates the version of any sequence revisions. Keywords, this introduces a list of short phrases assigned by the author describing gene products and other relevant information about the entry. Source, it provides information on the source from which the data has been derived. Sub-keyword organism illustrates the biological classification of the source organism. Reference, this indicates the position of the sequence data to which the cited literature refers. Sub-keywords include authors, title, journal, medline and PubMed pointers, etc. Comment, a free text section that allows any additional annotations to be added to the entry. Features, marks the feature table whose purpose is to describe the properties of the sequence in detail, such as the gene name, coordinates of the coding sequence. Database cross-references made through the /db_xref qualifier and to its sequence translation in GenBank's peptide database, for example GI 190470. Base count details the frequency of the different base types in sequence. Origin, this notes the location of the first base of the sequence in the genome. Then the nucleotide sequence itself follows and the entry terminates with ##. To access to GenBank as to all databases at NCBI is through the Emmrc search program. This front-end search interface allows a great variety of search options. More importantly, it enables searching more than one database at the same time, interpreting the search parameters in an appropriate way for each database. Here you can see the home page of Emmrc search engine. Recently, the NCBI Emmrc search engine has been modified as gQuery, NCBI Global Cross Database Search. Emmrc is NCBI's primary text search and retrieval system that includes DNA and protein sequence, structure, gene, genome, genetic variation and gene expression. It includes about 40 molecular and literature database, and these linked databases are listed as shown here. Biosystems, books, conserved domain, DBGAP, DBWAR, epigenomics, EST, gene, genome, genome project, GEO datasets, GEO profiles, GSS, homologene, MESH, NCBI website search, NLM catalog, nucleotide, OMIA, OMIM, PopSet, probe, protein, protein clusters, PubChem bioassay, PubChem compound, PubChem substance, PubMed, PubMed Central, SNP, SRA, structure, taxonomy, UniGene and UniSTS. To submit sequences in a database, submission tools are available. BankIt is a worldwide web-based sequence submission tool which can submit sequence directly to GenBank. Another tool, Sequin, developed by NCBI, is standalone submission software. Sequin can also be used to submit and update sequences in DDBJ and EMBL. A user can submit single short sequence, long and complex submissions, segmented sets of DNA, phylogenetic, environmental, mutation, population studies and multiple annotations with Sequin. Let us now summarize what we have discussed so far. Databases are electronic filing cabinets to store vast amount of information from research areas including genomics, proteomics, metabolomics, microarray gene expression and phylogenetics. Thus, they help to understand the biological phenomena from the structure of biomolecules and their interaction and the whole metabolism of organisms, and to understand the evolution of species. Based on the information they contain, databases may be primary or secondary. Primary databases contain data derived experimentally, whereas secondary databases include data obtained from the analysis of primary databases. Databases are also classified in two: sequence, structural and specialized databases. Based on the information we need, one can choose the best database that provide the needed data. Also, we can upload information into these databases using various tools. Now, you can try to answer the questions given here. 1. Explain the relevance of biological databases in bioinformatics. 2. Briefly describe the different types of sequence database. 3. Describe Emmrc search engine. 4. Explain the primary nucleotide sequence database. Now, you can go through the reference books for further reading. Bioinformatics, written by Darbeshwar Roy, published by Narosa Publishing House Private Limited, 2009. Bioinformatics, Databases and Algorithms, written by Gautam N, published by Narosa Publishing House Private Limited, 2006. Hope that you enjoyed the session. We can meet again with another topic. Have a nice day.
Hello and welcome to the session on styles of programming in programming instruction. In the last unit, we have learned about program learning or programming instruction. As seen already, in this method, instructional materials are presented to students through small units of information called frames, which are arranged in logical sequence. This method gives students immediate feedback and reinforcement regarding the correct answer at each step as they proceed from one frame to another, which in turn promotes self-paced and individualized learning experience. The process of arranging the subject matter in a progressive and logical sequence is called programming. Two major styles of programming are used in programmed instruction, namely linear programming and branched programming. According to some experts, there is a third style named mathematics programming. In this unit, we will take a detailed look at each of these programming styles, their features, relative advantages, limitations and procedures. By the end of this unit, you will be able to define linear programming style, examine the principles and theories underlying linear style of programming, identify the main features of linear style, gather basic understanding on how subject matter is arranged and presented in linear style, critically analyze the advantages and limitations of linear programming, define branched programming and its chief characteristics, analyze the major steps involved in branch reprogramming. Understand the two subtypes of branch reprogramming, namely forward branching and backward branching. Learn the advantages and limitations of branched style of programming. Define mathematics and its characteristics. Compare and contrast these different styles. Let us start with linear programming. Have you heard of it? Linear programming was developed by an American psychologist, behaviorist and social philosopher named Burrhus Frederick Skinner, popularly known as B.F. Skinner. Hence, this style of programming is also known as the Skinnerian style of programming. It draws from Skinner's theory of operant conditioning, which was developed based on the work of Edward Thorndike. According to this theory, it is possible to condition and shape the behavior of an individual step by step with suitable reinforcement in each step. Operant conditioning, also known as instrumental conditioning, is a type of learning where the behavior of an individual is modified by its antecedents and consequences. This theory emphasizes on the importance of reinforcement. In other words, a behavior tends to get reinforced and strengthened if it is repeated. A behavior which is not reinforced tends to get died out or extinguished. In the context of education, this theory means that students can be led to a desired behavior through a carefully designed program and by giving reinforcement in the form of a favorable experience or reward. Learning takes place when a reinforcing stimulus is presented to students to reward the correct responses they give at each step of instruction. Would you like to know the basic steps of linear programming? In linear programming, students move from one frame to another directly and they complete all the frames in a linear or straight line fashion. Let us examine some of the basic steps used in this style of programming. Firstly, the subject matter is defined and is broken down and arranged in a number of meaningful units or elements called frames. These frames are presented to students in a logically arranged sequence. It is important to note that the frames are presented to students one at a time. Such a pre-arranged and step-by-step presentation of material helps reduce the error rate. Once a frame is presented to students, they need to actively respond to it by providing their responses to a question. At each step, students are given enough information to make sure that they make the correct response before going to the next step. Immediately after responding, students are given feedback regarding the correctness of their response. The immediate confirmation helps students proceed at their own pace and check their own progress. This act of giving immediate feedback helps to reinforce the behavior of students. Moreover, they feel motivated to learn the next frame in the sequence. Thus, students proceed from one frame to another, and at the end of instruction, they are able to achieve the desired learning experiences and the terminal behavior. The sequence in which the frames are presented is linear and systematic in nature. That is the reason why this style is called linear programming. Since students enter the instructional process with their entry behavior and proceed towards the terminal behavior following a straight direction, linear programming is also known as a straight line programming. The whole procedure of instruction is controlled by the programmer and the nature of control is extrinsic. Therefore, linear programming is often referred to as extrinsic programming. Linear programmed material consists of carefully constructed questions and answers related to each simple item. The primary aim of this style is to make the sequence of response errorless. To achieve this, the size of each step is kept very small and they are sequenced in such a way that students make only correct responses and consequently receive only positive reinforcement. If they make an incorrect response, they may either have to repeat the frame or they are given the correct response. Students are not allowed to move to the next frame unless they respond correctly to the present frame. Now we are progressing to discuss the characteristics of linear programming. As already mentioned, this type of programmed learning follows a linear arrangement of frames. Following are the main characteristics of linear programming. The path of sequence and responses are controlled by the programmer, leaving students with little or no choice of their own. Emphasis is given to the responses of students. If learning has to occur and the process of instruction has to move forward, students must respond to each and every frame. In other words, a new frame is presented only if students respond to the previous frame presented to them. This type of programmed learning gives immediate feedback to students. They can compare their responses with the response given by the programmer. Thus, students can know the results immediately after the responding. Students are given prompts at each frame which help them respond correctly to the questions. Though the act of prompting does not promote learning, it helps to ensure that there is no wastage of time and students move from one frame to another at their own pace. Moreover, linear programming has a simple mechanism and structure compared to branched programming and methodic programming styles. Is linear programming free from any limitations? No, it has some limitations of its own. Firstly, linear programming is likely to make the process of learning monotonous. Since it follows a linear sequence and the frames are very small, a lot of time is spent to teach simple points and basic concepts. Another limitation of linear programming is that the serial order followed in this method might not necessarily correspond to real life situations. That is to say, linear programming presents information to students in a serial order. If students get excessively conditioned to the serial order, they might find it difficult to learn when materials are presented to them in a non-serial order. Thirdly, in linear programming, students don't have any role in discovering the answers, but they follow the path prescribed by the programmer. Such a lack of choice in responses might limit the creative imagination of the students. Linear programming is ideal for subjects where facts and information can be properly sequenced. For example, it is best suited for science, mathematics and vocabulary development in languages. However, the type of programming has limited or no scope when it comes to subjects where information cannot be properly sequenced in a linear fashion. This style of programming cannot be used for remedial purposes. It is suited only for teaching the subject matter. You have already learned in detail about linear programming. Now we are proceeding to another important type of programming, that is branched programming. Branched programming was developed by an American psychologist named Norman A. Crowder, who was a contemporary of Skinner. Hence, branched programming is also known as Crowderian style of programming. Crowder developed branched programming in the 1950s for the US Air Force. Though the primary purpose of this style was to train troubleshooters to find malfunctions in electronic equipment, it was later adapted for educational purposes. Crowder defined branched programming as one that adapts to the needs of the students without a medium of an extrinsic device such as a computer. Unlike linear programming, it has an intrinsic arrangement and the responses are not controlled extrinsically by the programmer. This means that students are free to make decisions and are able to adapt instruction to suit their specific requirements and needs. This is the reason why branched programming is also known as intrinsic programming. Now let us examine how branched programming carries out the process of instruction. Like in linear programming, here also the learning materials are arranged and presented to students in the form of frames, but the frames used in branched programming are of relatively bigger size. Branched programming is based on the assumption that students can learn better if the subject matter is presented to them in totality or in the form of meaningful components. The frames, however, are not presented to students one after the other in a linear or straight line fashion. It is the response given by students that determines which frame they should see next. Branched programming has two sequences of frames. One is the main teaching frame, where students are presented the frames one after the other. The other is the remedial frame, where students are directed to the next if they answer the questions incorrectly. Each frame contains two or more related ideas. The responses of students are collected by asking multiple choice questions. Students have to choose the correct response from a set of alternatives given to them. If students answer the questions correctly, then they are taken to the next frame in the main teaching frame. If they answer the questions incorrectly, they are directed to the remedial frame, where their mistakes are explained. Depending on how students proceed in the main and remedial frames, branched programming is further divided into two types: forward branching and backward branching. Let us take a look at these two types in some detail. In forward branching, whether students make correct responses or not to the question presented in one frame, they are taken to the next frame in the sequence. If the response is correct, students are directed to the next frame in the main teaching frame sequence. If the response is incorrect, they are directed to a remedial frame. The mistakes made by students are explained in the remedial frame, and from there they are taken to the next frame in the main sequence. That is, if students make correct responses or incorrect responses, they will be physically progressing from one frame to another. Whereas in backward branching, students are to repeat the frames for which they provided incorrect responses. If the response is correct, they are taken to the main frame. If students make an error in the response, they are taken to the remedial frame to help them understand the mistakes. After this, students are directed from the remedial frame to the original frame, that is, the one in which they provided the incorrect response. They are to read the original frame once again and attempt to answer a similar question in the light of the remediation they have received. Thus, if students make incorrect responses, they have to go through same frame two times, one before remediation and one after remediation. Therefore, this type of branched programming is called backward branching. Like linear programming, branched programming has some special characteristics. In branched programming, frames of larger size are used. Likewise, more information is presented to students at each step. Multiple choice questions are asked to confirm whether students can respond to questions correctly. The sequence of frames is not determined for all students by the programmer. The material presented to students is determined by their performance and knowledge in the subject matter. The path of sequence is determined by the correctness of their response. Next, errors are detected and rectified at each step. This style of programming gives emphasis to problem solving. This style is based on the assumption that learning occurs better when students are exposed to new material. Would you like to know the various advantages and limitations of branched programming? Following are the advantages of branched programming. The use of larger sized frames and the branching structure help to avoid unnecessary repetitions and responding. This also helps reduce the learning time and makes learning less monotonous. Instead of simple responses, this style uses multiple choice responses with alternatives. Moreover, it helps students to rectify their mistakes as the errors and mistakes in the responses are explained in the remedial frame. It helps in the development of a student's power of discrimination. It also promotes creativity and problem solving skills. Additionally, this style can be used for teaching subject matter which is beyond facts, definitions or basic skills. Branched programming is with some limitations too. Since this style uses multiple choice format, there is an increased risk of students guessing the correct answer without understanding the material presented in the frames. It is difficult to determine in advance how many branches a student will require. Therefore, branched programming does not cater to the needs of all students. Moreover, when long frames are used, followed by several questions, it will look more like a general textbook. Branching style cannot provide infinite branching to take care of all possible needs of individual students. The cost of designing branched program is higher compared to traditional instruction methods. Also, it is not suitable for students of lower grades, but is appropriate to be used only in higher stages of learning. Since the frames are large and contains more information, it is not possible to ask questions that cover all aspects of the content matter presented in one frame. Hitherto, we have seen the principles, assumptions, procedures and characteristics of both linear and branched styles of programming. Based on this, can you identify any fundamental differences between the two? In linear programming, all students go through the same path in the same linear sequence, but in branched programming, students follow a path according to the response they make. The frames used in linear programming are relatively of less difficulty, but the difficulty level of frames used in branched programming is comparatively high. Similarly, linear programming frames are easier to construct compared to branched programming. In linear programming, the frames are smaller in size, but the frames of branched programming are big in size and contain more information. Linear programming offers no remedial instruction, but branched programming has remedial frames where the mistakes made by students are explained in detail to help them rectify the errors. Linear programming is extrinsic. The path to be followed by students is determined by the programmer, but branched programming is intrinsic. Linear programming has to construct responses while branched programming uses multiple choice questions. The third style of programming used in program instruction is methodic programming, which was developed by Thomas F. Gilbert. He defines methodics as the systematic application of reinforcement theory to the analysis and construction of complex behavior repertoires which represent the mastery of subject matter, knowledge and skills. The word methodics is derived from the Greek word methein, which means to learn. The procedure of this programming is as follows. It begins with an instructional plan and a detailed analysis of what is to be taught. The unit of learning in methodic programming is not called a frame, but it is called an exercise. There are no restrictions regarding the size of the exercise. The size of the exercise is usually determined by what students can reasonably handle at each step. The reinforcement value of accomplishment is given emphasis in each exercise. This style is based on the principle that the closer students are to reinforcement when they are taught, the more effective the reinforcement will be. Here, the term reinforcement does not refer to knowledge of results, but it refers to the completion of a task. As we have already seen, other types of programming follow a pattern of arranging individual frames in a logical manner. Methodic programming, on the other hand, analyzes the type of task involved in mastering the content. In other words, it does not give too much importance to the logical structure of units. The sequence in which exercises are arranged in the methodic style is quite flexible compared to linear and branched programming. It is important to note that the sequence may differ from task to task even in a single unit of instruction. This style emphasizes the fact that motivation is an important factor in the completion of a task. one of the most important characteristics of methodic style is retrogressive chaining. What gives meaning to students is usually the last task or the completion of the task. Therefore, methodic program begins from the last task in the sequence and moves backwards. Students complete the last task first and then they go to the next one till they reach introductory part. This procedure of moving from the last exercises to the first is called retrogressive chaining or backward chaining. Let's move to discuss the advantages and limitations of methodic programming. The major advantage of methodic programming is that it is task oriented. Moreover, the results of the program can be linked to the instructional goals and objectives that are expected to be achieved through the program. Since this style emphasizes the success of the learner, it keeps students motivated in the process of instruction. However, this style has a number of limitations too. The technical nature of this style demands a lot of time, labor, training and skills on the part of the programmer. Also, it cannot be used with the learning material of all subjects. The individual differences of students are not given adequate attention to. Methodic style can be used neither for factual content nor for higher cognitive and affective objectives. Moreover, this style does not give remediation for incorrect responses. So, the weaknesses and learning difficulties of students are not addressed properly in methodic style. Now, you can try to answer the questions given here. What is operant conditioning? What is its significance in the context of education? What is forward branching? What is backward branching? What are the major limitations of branched programming style? Mention at least 3 major differences between branched and linear styles of programming. What is retrogressive chaining? Hope that you may go through the reference books given here for further understanding. Essentials of Educational Technology, written by Agarwal J.C. and published by Vikas Publishing House, New Delhi in the year 2009. Sociology of Education, written by Chandra S.S. and Sharma R.K. and published by Atlantic Publishers, New Delhi in 1996. Teaching of Social Studies, written by Mangal S.K. and Mangal U. and published by PHI Learning, New Delhi in the year 2008. Essentials of Educational Technology and Management, written by Mittal M.L. and published by Pearson Education India, New Delhi in 2012. Teacher Education, written by Mohan R. and published by PHI Learning, New Delhi in 2011. Education Technology, written by Pathak R.P. and published by Pearson Education India, New Delhi in the year 2012. Introduction to Educational Technology, written by Sambat K. and published by Sterling Publishers, New Delhi in 2001. Introduction to Education Technology and ICT, written by Sheikh I.R. and published by McGraw-Hill Education, New Delhi in the year 2013. Thank you for watching this program. We can meet again with another topic. Have a nice day.